All right, good morning, everyone. I hope you are all having a lovely Saturday. Hello and welcome to Science Rendezvous 2022. So my name is Carrie and I will be your MC for webinar 2 of the SR show. We are very excited to have you all join us for our second virtual festival, and we look forward to seeing some exciting presentations from our special guest speakers today. So to start us off, I do want to give a quick introduction of the SR team here at webinar 2. Like I said, my name is Carrie. I will be the MC for today, and helping us here are Helen, who is our community engagement, and she runs programming here at SR. So she's been super involved in getting the whole program done as well. We also have Presley joining us to help out with community engagement across the whole webinar. Taking on the Zoom production is Henry, who will be helping me go through all of these slides. And we also have Jenny, who will be kind of going through the Zoom chat, as well as helping us out if there are any issues. And Miranda is also here joining us as our social media lead. All right, so that's our team for today, and we look forward to having a great webinar here in room 2. All right, so for our agenda today, please do visit the program schedule here at the Science Rendezvous website, and you'll be able to see the list of presenters from 10 to 5 p.m. So our website here is going to be the one that you see here, but it's www.sciencerendezvous at uoft.ca. And if you go under the 2022 festival in program, you'll be able to see the whole schedule and kind of figure out where you are on the day. So before we begin, since we are doing this virtually, here are a few best practices to please keep in mind as you go through the day. Firstly, please post in the Zoom chat. Our presenters, when there is time at the end, will be taking questions during their segment. And secondly, if you do have any technical issues or questions relating to logistics throughout the module, please do send your questions to the email listed here. So it's uoftsr.virtualhelpdesk@gmail.com. If there is an emergency, you can go ahead and email them, and we'll be able to kind of help you out there. Okay, so before we do start, we do want to give a big thank you to our partners, because without their support, Science Rendezvous would not be possible. So big shout out to all of these partners who have been helping us through the whole year this year. All right. So Stanley, how do you feel? We can kind of give a few more minutes and see if people trickle in, and we'll give you the floor at 10:10.
Sounds good.
All right, perfect. Thank you. So I'll do a quick introduction at 10:10 and we can get started. It's a really exciting day. All right, so we have Stanley joining us today from Near and Middle Eastern civilizations, who will be our first presenter, and he will be kind of going through the crane and the TAP projects with us this morning. And as a reminder for all our attendees, if you do have any questions for our speakers, please post it into the Zoom chat and we will answer them if there is time. All right, Stanley, whenever you're ready, if you want to share your screen. Beautiful, we see it. Wonderful. Okay, all right, let me give you the floor and give it up for Stanley. Woo.
Thank you, Carrie, for the introduction, and welcome everyone to this Science Rendezvous, and it's a pleasure to be here as always. My name is Stanley Clausen, and I'm the collections manager and lab technician in the archaeology lab at the Department of Near and Middle Eastern Civilizations. So today I want to tell you a little bit about some of the projects that we're involved in. TAP stands for the Tainat Archaeological Project, and CRANE is for the computational research on the ancient Near East. So Tel Tainat is situated in the Hatay region of Turkey, and that is one of the U of T projects that that I'm working with, and we are heavily involved with the CRANE project, which is a partnership grant. You can see the list of many of the partners at the bottom. This is funded by the Social Science and Humanities Research Council, and you can see there's another sponsor that was the ROM is also part of our one of our partners, is also a sponsor of the Science Rendezvous, which is nice to see them participating in overall as well and supporting this Science Rendezvous again this year. So you can see a number of the universities and other institutions we have. We are not just University of Toronto, it is an international collaboration, and we've been at this for over a decade. We are now into CRANE, we're calling CRANE 2.0. We've gone through a first round of funding, which is a 5-year grant. We switched into 6, and now we're into the second round of funding. So we have moved from our initial data integration and expansion in the Orontes watershed, and we are now moving into some other exciting areas, which I'd like to talk to you about today. So here we go. So again, just a little background of where we actually work. So you can see in the map here, we are working in the Orontes watershed. That is where the original project kicked off. And you can see Tel Tainat. I don't know if you can see my cursor, but Tel Tainat is just near the ancient site of Antioch, which is Antakya in the Hatay region of Turkey. But you can see the Orontes snaking up towards Tainat and then heading over to the coast. So that is the region that we've been working with originally. It's basically the watershed draining over that 24,000 square kilometers, and it has a very rich archaeological history in the region. And you can see here just a bit of a three-dimensional look. You can see the Amanus Mountains in the background with or Zingerli. Ancient Samal is towards the north. And you can just see over the tip of the Amanus Mountains, you can see the Mediterranean Sea there. So this is the region that we're actually working in. And this is our initial. We are now expanding well beyond this original Orontes watershed region into parts of Lebanon, and also Palestine, Israel, and parts of Iraq as well. So we're really, and Jordan as well. So we're really expanding the breadth of our research into not only the northern Levant, but into the southern Levant as well. Again, hats off and kudos to all the students and researchers that we have here at University of Toronto. This is part of the project that I'm heavily involved in, is actually the data integration of all the various sites and the data that we have from the various projects that we're integrating into the larger database. And we work here at University of Toronto. We do have a lot of work study students and researchers. So if and when you come to University of Toronto, we do have work possibilities to help out with the CRANE project as well. So it'd be wonderful to have you on board as part of that project. Just an example of some of the students that have worked with us over the years. We've had over 25 students working with us, and we've invested in the data integration probably in excess of 10,000 hours of work that we've employed students so far. So just here's a few of the projects. You can see that we've actually are part of the data integration process. Tel Tainat, of course, is what I will be talking to you a little bit about today, but we've also got Ancient Samal, which is Zingerli, and there's a few other ones. The ceramic repository, the Orontes watershed is a big one. Crow, we do like bird acronyms, so it's Crane and Crow, and as well as some other legacy projects, the Gezer excavations, and also Wadi Tumlat, Mastuma, and Tel Arka in northern Lebanon. So we've got a lot of these projects that we've been working with that are now on board, and we've continued to expand our geographical region. The types of resources that we're actually integrating into the larger data set that we can actually use for our research are things like rectified images, GIS imagery. We've also got locus sheets, field notebooks from the various excavations, along with objects, whether they're pottery or other artifacts, field photos, aerial photos. So we have a large amount of data that we're actually integrating into it. And just to look at some of the numbers, this is a little bit out of date. It has 122,000. We are probably in excess of more like 180 at this point. So we've got an awful lot of resources that we've been integrating into the larger data project. So we can, and I'll show you some examples of how these actually work in our data set. So I want to again talk a little bit about TEL-TINOT, which is one of the projects here at University of Toronto that is a key component of CRANE. So the name of the site is Ancient Kunalua, and it was excavated in the 1930s by the University of Chicago. You can see the black and white images on the left, some of the excavations images from that time period, and we've initiated new excavations, and you can see there's an aerial view of TEL-TINOT. That is just the upper mound. That what's visible there, that sort of brown spot with some current buildings, some factory that was built. But you can see the Orontes snaking in behind there with in the highway splitting between the Orontes River and the TEL itself. So we'll talk a little bit about the Orontes and how that actually comes into play in our understanding of the region through time. One of the very interesting finds that we had is some very interesting monumental sculptures. And we've also excavated some temples with some tablets. So here's just a couple of images of some of the things that we've excavated as the University of Toronto project. So we'll look at a little bit more of that as we go through here. So we can see Shupi in the top right. We'll focus on him in a little bit. And we have that beautiful lion. Both of those are about 2 tons made of basalt. And they are about 2 tons in weight. Very, very beautiful finds. So here's the TINOT temple that we excavated a number of years ago. Went through a number of phases, but you can see the mud brick architecture with the stone paving, the steps leading up in the paving inside. There's a GIS architectural features are shown in the larger print on the right-hand side. And this is the excavation of major find that we found is the tablet. And we found a number of cuneiform inscriptions. So this is written in a Neo-Assyrian language called Akkadian, and it is actually dated to SRHaden in 672 BCE. We actually have the date right on the tablet. So this is the extraction of it with our director, Tim Harrison, and our Julie Enro is our conservator. A very painstaking procedure, but there you can see the tablet in situ and rather fragmented, but Julie Enro has done an amazing job to conserve that tablet. Here are some of the other finds that were found in the back of the temple. We got a nice, beautiful little S.T.E.A.M bowl, a little bit of sort of a gold leaf coil, and some other metal objects. And of course, we have a lot of pottery. This is an Assyrian glazed ware piece, and you can see a little bit of the texture and the color, sort of a bit of a yellow glaze on the exterior. But you can see the image of that animal with sort of a lotus blossom that's on the side there. So that's depicted on that bowl itself. So this is the tablet, which is conserved now in the Antakya Museum, in the Hatay Museum in Antakya. You can see that glazed bowl in the back to the right. So this is the reconstructed tablet, and it's written on both sides. And as I mentioned, it's dating to 672. It actually has the date right on, and it's a treaty between Esarhaddon, the Neo-Assyrian rulers, the ruler of the time, and the local rulers in the region at ancient Kunalua. And it's a treaty document, very similar to one that was found at Nimrud in Iraq in the 70s. This is the sculpture of the lion, the 2-ton lion that we found buried below, and you can see it was tossed over on its side and buried with stone paving over top of it. A very difficult lion to move. You can see here it is being loaded onto the truck and then carted off to the museum in Antakya. The truck was too big to actually get through the gate, so the line needed to be hoisted up over top of the fence into the compound itself. But a very tricky, somewhat unnerving move that we got the line intact. But I mean, the line is now intact in the museum, and there I am standing beside the line in the Antakya. You can see the size of the line, 2 tons. It's a beautiful piece, completely intact, and interestingly, one of the pieces found by the Chicago excavations at another temple just to the left of our Temple 16 is 2 lions with exactly the same sculpting technique. So the very same sculpting school, and that is a pillar base. So you can see that they're very much dated to the very same time period. Shupi Liliuma was found the next season, you can see the individual face down, right in, you can see the temple in the background. Here we are excavating, and we know it's Shupi Luliuma based on the Luvian inscription on his back, which says, I, Shupi Luliuma, did this, that, and the other thing, of course. I mean, talking about their exploits is very often common on these types of sculptures. So this is an amazing find, again, approximately 2 tons, broken at the waist. Here he is getting cleaned up and transported on that crate, moving to the Antaki Museum. And here I am again with Shupi in the museum. So you can see the amazing sculpture that we have found, broken at the waist. We're not certain if he was seated or standing, but a very monumental piece of artwork with intact inlaid eyes. So a really remarkable piece that we found. There's Shupi face down with a few other objects. There's another capital, the pillar base. And we've also got what we think is a bit of a gate complex, and you can see the paving there. So this was found right in front of the temple. In 2017, we were able to return and we excavated again a little bit to the left of where we found Shupi, and lo and behold, we found another monumental sculpture. And when we removed and lifted this individual up, we realized that it was in fact a female. And here you can see Shupi and the lion in front of the temple, and just to the left, the Lady of Tainot. That small, what looks like a fairly small sculpture is actually quite large. But interestingly, she was defaced. Unlike Shupi, the Lady of Tainot was actually defaced. So we found thousands of basalt artifacts. You can see them in the image at the Antaki Museum in the storeroom there. We're actually trying to reconstruct and see if we can piece some of these items together. So that is the Lady of Tainot that we found there as well. Now, just to go through some of the databases, when we find all these artifacts and all the field records, we are uploading these into a database that we call called Ochre, and we can actually work our way through and clicking on the various images, we can see not only the images, the field records, but the locus sheets, all the information. We can click on the particular find spots, the artifacts, all the details that are involved in each of these type of items, that whether it's, we have the drawings, the measurements, the types of materials they're from, and the same is true of pottery. So we can have all the different attributes, the shape, the size, the fabric, what it's made of, all the details are a large part of that database. And the great thing about the database is it's searchable, not only within the site itself, but we are now expanding through, we can now search for similar types of artifacts in our database throughout the entire Orontes watershed region, and now we are expanding well beyond that. A big part of our project is looking at the regional mapping. Here are some of the sites. We've got over 4,400 sites you can see that we've got documented here. To identify these, we look at Google Earth, different type of raster, GDM, corona, and topographical maps to clearly identify specifically and exactly where these are located. One of our projects is working with the Syrian Heritage Initiative. During the conflict in Syria, we were actually documenting where sites were and if they were destroyed, we were able to document a lot of these sites, and we've added a huge number into the larger database of the historical and cultural heritage of that region, which has been really, really important due to a lot of the destruction that has taken place in the region over the last decade or so. Here again, just to look at all the dots are particular sites that we have documented. So there's an awful lot of sites. One of the other projects we call Fueling the State. So this is coming out of Durham University with Kamal Badrashani heading the charge on this one. So what they're actually doing is looking and investigating how ancient pottery was produced and the types of fuels, doing experimental work. And the recent work that Kamal is finding is actually finding that olive pulp was probably one of the most used fuels that was being used to fire the pottery. So again, as they're pressing the olives, adding the oil to the various jars, they could actually burn that pulp in packed cakes, and it's a very useful fuel. Just some of the fuel testing they've done. I don't want to spend too much time on this, but one of the key factors is also looking at experimental potting. So trying, as we understand the types of ingredients and the minerals and the inclusions, the clays that are being used, we're testing those and seeing exactly how these types of vessels were produced. So I have just a little video here, and you can see, if we can get it running, there we go. So we've got an experimental potter who's actually making pottery. I'll just zip through here. You can see they're actually producing pottery that we have found. So they're recreating some of this to really understand the technology and get an appreciation of how these vessels were made. The types of inclusions we're adding is very similar to really try to reproduce and get a better understanding of the production and of these ancient vessels that we are finding. So I'll just advance here quick, and you can see there she's working on it, and the final product is one of our storage jars that we've found. So it is an exact replica of what we are actually dealing with. Okay, another thing we're looking at is 3D visualization. So we often find sherds or partial sherds, and we are able to draw those in two-dimensional, but we're actually doing using three-dimensional scanning, and we can actually recreate and try to place these artifacts in a three-dimensional image, get a better understanding of the actual excavations. And we're also looking at color, how that plays into fact. A lot of these buildings would have been maybe only have maybe lamps would have been the fuel and the lighting and maybe some window wells. So we're looking at how color plays and how difficult it would be to see those types of colors and how that interacts with the use of these vessels in very dark spaces. So there's lots of interesting techniques working with computer science here at University of Toronto. I've been leading the aspect there. So again, here's a two-dimensional drawing of this little crater. And what we're able to do is running it through various types of software. We can recreate it in a three-dimensional realm. And you can see here, and there's the vessel completed using this software. So using 3D modeling. So we can then actually place these types of vessels. There's our glazedware vessel that we found at the S.T.E.A.M in the temple. We can then place these vessels in various formats in gaming, which we'll talk about, gaming modes, and we'll talk about that, reconstructing some of the ancient world that we have. 3D matching is a huge component that we are really sort of kicking off, looking for the thousands of mens that are the potential mens from the various Luwian inscriptions, but also trying to reconstruct the lady. Again, here she is. You can see, remember, she was her face was defaced. So we've actually been able to find a few men. So there is the lady as we have been able to reconstruct her. So using some of the parts, we are continuing to looking for more basalt images or pieces that we can actually add to her and get a sort of try to recreate what she would originally have looked like. 3D modeling and visualization, as you can see with the pottery and the artifacts, is a very big component as a project trying to develop and use this for modeling and for outreach as well. We've looked at various types of gaming modes. We again are looking at the two-dimensional space that we're working with from our drawings and converting that through various types of things like SketchUp and other types of software. We can see we've got the model plan in GIS, and then we can move into a more of a three-dimensional. So from the conservative to the imaginative, of course, and then we were also looking at some of the 3D modeling and creating the actual towns based on our excavation and our knowledge of the site. So here's some of the graphics that we've been able to produce of what the site probably would have looked like based on our excavation and the accuracy of our the historical data we have. There is the temple that we talked about at S.T.E.A.M. Again, there's our GIS, and we can see how we've gone from the GIS images through SketchUp into gaming engine. So trying to recreate the temple as it would have and perhaps looked like based on our excavations and the information that we've uncovered. We can also look at various landscape and based on our paleobotany and the faunal analysis that we've been able to determine the species of plants, we can then recreate the environment using these types of gaming modes. And so this is a lot of work done by Stephen Bacek. So we can add trees, vineyards, some of the buildings, olive groves, and forests in the mountains based again on the species of the carbon material that we've been able to find. Another project that we've been working on is coming out of Central Florida. This is called DATCH. So they're using virtual reality goggles, and they've created software where you can actually you have a menu within the screen. They're still at the prototype stage, but they're moving into the full application of these goggles, and you can actually draw. As you can see the image on the left-hand side, there's a young girl who's actually drawing with the visual graphics the walls and the architectural features within that, and then that is recorded as well. Again, so the interactive augmented reality is a big part of our work as well. We've got a group in UBC that is working on a Cypriot project here. And again, the basic excavation is in sort of two-dimensional, and there's the pottery, and you can see where it was actually found in the particular rooms. That's what the site looks like today. And there's some of the large vessels that they've actually had uncovered, and some tomb material as well. But what we're trying to do then is recreate that using virtual reality and allowing people to visualize what it would have looked like based on the artifacts from the past that we've uncovered. So that's spearheaded by a project out of UBC. So you can sort of see there, that's the virtual reality, the 3D building on the right-hand side, and the pillared building as it looks today. Ideally, we're hoping as people visit the site, they will be able to look at this through their phones using QR codes, and we'll be able to then visualize on site what it would have looked like in more of a three-dimensional space. So we're really trying to bring that 3D world to what is in a more completed sense to what is actually seen at the excavation on site. Key to our project, of course, is building the regional chronological framework. So this is based on C14 dating and using a high resolution absolute chronology, not relative chronology, which is more based on pottery, but this is really getting specific dates. So it's based on dendrochronology and also carbon-14. There you can see Stuart Manning. This is coming out of Cornell University. So they're looking at, and Bria Lorenzen is actually one of the lab assistants who's been working with Stuart, really developing our type chronology for the region using dendro and also C14 dating. There's just some of the work that they're doing. So how they determine the dendrochronology is actually looking at local trees. Some of them are hundreds and hundreds of years old. Also architectural features where they are able to core and look at tree rings and create a large and a broad date for the region. Here's some of the species, what they look like under the microscope. And what we also are doing is trying then using that data and coring. You can see these individuals here coring in parts of Syria, are looking at the paleo-environmental reconstruction. So trying to determine what did the environment look like in the past and how did it change? Why did settlements move from one area to another? So we are also looking at isotope analysis. So here you can see where we're looking at the water availability during the plant growth. In some would show water stress, or in some cases, different nutritional values. So we can see as these are short-lived samples, so based on one crop harvest. So we were actually be able to tell during that harvest what the environment would have looked like during that time using the type of isotope. So the Department of Physics here is looking at global climate modeling. They're one of our partners. They're looking at sort of larger global climate models, and you can see the resolution is 100 to 200 square kilometer grid is normally what they would use. They are fine tuning that, and we're getting down to the point we're looking at what's happening within a 30 square kilometer or even down to a 10 square kilometer resolution. So you can imagine in places like Toronto, you've got sort of that lakefront effect. What's happening along these, along the lake, and what's happening in places like, say, Barrie or in Hamilton, very different kinds of environments, even though you're looking at a very small environment. So that's true of the ancient world as well. So we're able to try to understand what is the climate and what sort of resources were available and how they interacted with their environment. Here's just a couple of images you can see. We got very dry, arid. We've got very humid, marshy lands, and also plains where agriculture products are harvested. So there's a lot of different environments, and we're really getting a clear understanding based on our type of research. So factors affecting agricultural productivity are quite wide, and our data and the results from a lot of our analysis is really fine-tuning and getting us a very good idea of the types of things that affect population and migration and movement of settlements. And we were trying to estimate the agricultural production of the region as well. Based on various types of proxy data, historical texts, a lot of the ancient sources talk about the different crops that are used, so we can use that information. So we have a wide variety of data that we can actually use to pull together. And then we try, of course, to reconstruct land use and settlement data within the region. And again, trying to focus more specifically on the regions that we look at. So again, all of this types of information that we are gathering through the various types of research is really pulled together to look at some larger questions and issues of how populations may have settled, where, why, and how they would have moved and types of environmental factors, even political factors are strongly affected by climate as well. So these are sort of the larger questions that we're trying to understand based on the data that we've been pulling together on our various sub-projects in the CRANE project. And if that wasn't all interesting and fun, we are also using Minecraft. So this is something that we've been working on as a sort of educational outreach to try to create, you saw the gaming models. I mean, these are often very difficult. The software changes quite frequently, but we were really trying to do is using Minecraft, which has been a very stable type of software and game that's really unchanged for decades now. So what we're doing is trying to recreate the ancient world in Minecraft, and we're well on our way to actually creating the world around Sci-Art and the larger region, and we're hoping to get that online so you as gamers and as interested historians and budding archaeologists can actually take a look at the ancient world. So you can see some of the outreach we've done here at the Royal Ontario Museum. Some of the things that we're looking is really learning about history and interacting. These are some of the individuals that we are going to be adding to the game. Again, this is all, it's very important that we're basing this all on historical documentation, whether archaeology or graphics. So we've got a few characters. We've got some NPCs created. There is Syro-Hittite Syro-Hittite scribe, Neo-Assyrian soldier. Of course, we have to have King Shupilululiuma. That's him there doing a little dance. And here are some of the other characters. As Minecraft develops, we are also getting more of the species, not only plants, but also the animals that were in the regions. We're not trying to create an unrealistic world. We're trying to make it as realistic as possible. And a lot of that's coming out of the 3D visualization at sites such as Carchemish, where we have these large reliefs that were found back in the 30s, and the documentation and the scanning that they've done, we're replicating the clothing and the imagery that we have for our characters to try to make it as accurate as possible. Also, things like drone imagery is also very helpful in determining the larger space and how we can understand the sites themselves. So again, from the 2-dimensional to the 3-dimensional, this is Tel-Tainot again. We can then create the ancient world in Minecraft. Here we are at night. And again, looking at the historical accuracy, which is very, very important. Some of the coring projects, you can see we've actually been able to determine that the sedimentation on either side of the Tel is more of a sand. It's not really alluvial or material culture. And interestingly, the Balawat Gate pictures Tel-Tainot on the inscription here, or on the gate sculpture. And it was always interesting because it looked like it was surrounded by water. But the coring actually that we've done is determined that Tel-Tainot at one time was surrounded by water from the Orontes marshland, or maybe even as much as a river there. So again, it's very interesting. We were able to go back and determine through our archaeological work. And of course, we now are recreating Tainot with the Balawat Gate. We still need to add the water for that picture there. But again, trying to imitate, you can see the temple there. We've got a pillar base in situ on the left, and we are now adding some of those textures to some of the basalt blocks in Minecraft to produce that. So hopefully we'll have the Lady of Tainot will be able to lead us around Tel-Tainot, and you'll be able to interact with some of the command blocks that we're creating and explore the region and learn about Tel-Tainot and region during the Iron Age period. Anyway, I'm going to wrap that up. If there's any questions, I'd be happy to answer. On behalf of the Tainot Archaeological Project and CRANE, I would like to thank you for your attention. And there are some websites. If you'd like more information, you're more than happy to consult those and see more about our projects. All right.
All right. Thank you so much, Stanley. That was absolutely incredible. I think it's always so cool when we get to see the interactions of using modern day technology to go back and studying the ancient world, especially, and so much cooler when we can see that there's like implications of like gaming in there as well. Yeah, so that was so cool. Let me pull up some of the questions. All right, so firstly, I think just a very general question for the program. What are sort of the things that you're looking for, or like the program is looking for, as well as what the project itself is looking for? So I guess that would apply for the Near and Middle, for your department, for
Right.
Near and Middle Eastern civilizations, as well as for the CRANE and TAP project specifically.
Yeah, so I mean, we're always keen to get back in the field, of course, due to COVID and travel restrictions, and we were unable over the last few years, but we are going to be back in the field this June. So we are very excited to get back to S.T.E.A.M. We'll be running a project. We'll be heading out at the end of June into August, early September. And what we want to do is expand the area, particularly around where we found the Lady of Tynot and Shupi, because we know and we're quite certain that there's more monumental sculptures, or at least pieces of those particular monumental sculptures. So we're trying to understand more about that time period. It was sort of local rulers ruled the area during the Iron Age and the Neo-Assyrian period, when the Assyrians came in and conquered the region. This is that tablet where we have that in the temple there, where SRHacks and his predecessors actually controlled the region and sort of set up a puppet state. So we're trying to learn more about the history of that particular time period, but we also have areas dealing with the early Bronze Age. So we're going to be excavating into what we hope is what we are quite certain is a massive destruction. It's visible on the ground, fired red brick superstructure. That is in the early Bronze Age 4 period. So that'll be dating to about 2200. So that's another area that we'll be excavating. And again, just expanding our research for that, and try to keep feeding more data into the larger database system to get a better idea of the region and how it interacted through the time periods, and how settlements moved throughout the region, and how people interacted with each other.
Cool. Super cool to get to know that. I know we have a technical question as well. When we were talking about the barley and the seeds, I think you mentioned isotope analysis. If you can kind of go, yes, if you want to go and explain a quick overview of what that exactly is. Why do we want to study isotopes? What do they tell?
The isotope analysis can be done on bone material, but also on faunal and also floral material. So carbon material and seeds are very important, because basically what we're looking at is particular isotopes that are telling, and they are drawn in from things like the limestone, the minerals where they are growing. We can identify the type of isotopes that occur in those minerals, and by looking at the isotopes within the seeds and plants, we can determine where they are growing, where they're from, and this is particular on things like faunal material. So we can actually identify that some of the say sheep and goat that we're finding on the site there, the bone material may be actually coming from another region. So looking at the isotopes of that type of material, we can look at location of where it's actually coming from. So we can start talking about migration when it comes to faunal material, but also the seeds themselves. We can really look at the type of nutrients and the rainfall. Was it a dry season? Was it a wet season? So all of that is really, because if we're looking at seeds that are short-lived, that is from one harvest. So rather than a tree which grows over a long period of time, is more difficult. We could only look at when it was cut down and used, but seeds themselves are from a particular harvest season. So having that kind of resolution is really, really important, and it's worth a lot of detail for our types of analysis. So there's a lot that can be done with isotope analysis. It's a very interesting field. I don't deal with that specifically, but I mean, we do have the scientists involved. So any of you, even though if you want to be archaeologists or even scientists, we are always leaning on other fields. It's a great collaboration. So I mean, you can always be involved in the project, even if you're very interested in things like science or engineering or architecture. We rely on a lot of these types of fields to help us out.
Yeah, the collaboration of science, definitely a key point. One other question we do have, Stanley, how did you get interested in archaeology in the first place? And has that changed over time?
Good question. Yes, I've always been interested as a kid, even, in history and, sort of, how people lived in the past, and I studied at the University of Saskatchewan. This is where I did my undergraduate degree, and I was taking sciences at the time, and I took a few courses in anthropology and ancient Near Eastern studies, and I realized that was really my true passion, and it just took off from there, realizing that those kind of courses were available, understanding the history of that particular region that I was so focused and interested in the ancient world of the Eastern Mediterranean. So coming to Toronto, I did other graduate programs, but it really seeing the availability of courses that focused on these types of things allowed me to continue and develop as an archaeologist. I've been doing this for decades now, working in primarily Turkey, Jordan, Palestine, Israel. But it blossomed, and as we continue and we becoming more adept at our techniques and how we actually excavate and the sciences involved, we are only getting better at this field, and it's just becoming more and more exciting as we go along. And the kind of detail that we can actually realize is just phenomenal. So I mean, it's something that I started out with as a kid, and I still absolutely love it. And I'm only becoming more enthralled in what I do and passionate about what we do. And it's been so much fun.
Yeah, that's super cool. I think that's definitely incredible. And for everybody here who are attending, I think that's like quite inspiring of a story as well, to really just find what interests you, right? Like you can start off from one point and just end up completely on, or not necessarily on the different end, but maybe not the same starting point as what you kind of go into school for.
Yeah, true. I mean, and starting with the sciences, I've now gone back into applying a lot of the sciences in the petrography that I do with the pottery and learning and mineralogy and geology and things like that. So I've kind of gone full circle back to it, and it's great that you can actually apply all of these different fields because it really is. I mean, to focus on it's multidisciplinary. I mean, it's really important to know that, and we really rely on a lot of collaboration, which is really exciting.
Yeah, so I know you talked about a lot of the sort of technology that we do use, and you're going back into sciences, kind of looking into and using all of these. What are you most excited for the future of archaeology, Stanley?
Wow, that's a good question. I mean, as archaeologists, we don't excavate a site to completion. I mean, that would be, we really want to get snapshots and look at certain areas that we are, we can identify through like the corona imagery and using radiometry, looking at ground penetrating technologies where you can actually identify what's below the ground before we excavate. So really sort of focusing on key areas that maybe have the best evidence or some of the best results, which is a time saving and a cost saving factor as well. I mean, it's difficult to simply throw plots down and not find anything, which often happens. I mean, you could end up in an area that is, maybe in the middle of a alleyway or something. So there's still interesting finds, but I think what is really intriguing to me is actually it's the climate modeling that we've been doing, and sort of, really realizing that what we apply to today's world in the modern world, understanding where climate's going, based on the research we can apply that to the ancient world. And really, I mean, we often say, when you look at the ancient world, it often we see repeats. I mean, the history does repeat itself. And so understanding the ancient world and where people lived, how they adapted to their environments, and really understanding all of those things, all that information informs us about ourselves. I mean, so I think really, and where we are going, how we adapt. So a lot of times we see a continuation, and some of the practices are very similar to where they were done in the ancient world, and how people react to environmental conditions and things like that are really interesting. So I think that is one of the most exciting things, is really seeing the potential in the future of the sort of the digital archaeology as well. I mean, you always have to have a spade in the ground. We're always working with excavations, but just the potential of some of the new technologies that are coming out there. It's just, it's so much is happening in a very short period of time that's really informing us quickly and allowing us to get this kind of information to the public and really letting people know and become passionate about our past. Really, this is really what it's about: understanding who we are and where we came from.
That's absolutely amazing. I'm so excited for that as well. I think that's a great note for us to kind of finish off too. The future of what archaeology would be. I think we're speaking to the future today as well. So that's super exciting. Thank you so much, Stanley. You gave an absolutely super exciting presentation about your work, your projects, the projects you've been involved in, along with other universities, and really delving into the deeper world of archaeology. So, I mean, speaking with you, I think your passion in your work is also very, very evident. So it's inspiring. I love it. Thank you so much for joining us today.
Well, thank you. It's been a pleasure to be here. Thank you.
Perfect. All right. Thank you so much, Stanley, again. And that was our first session. I would say that was an incredible start for us today. We will be going on to our second presentation for today, and that will be from the U of T stem cell group, joining us with hope stems from donors. So again, I will come on and remind everyone again that if you have any questions for our speakers, please do post it on our Zoom chat, and we'll be able to answer them if there is time. So let me give this to the presenters here, and all right. All right.
I cannot share this screen.
All right, Rupal, let's figure this out. Do you want to try again now? I think you were just given co-host.
Okay. Great. I can.
Perfect. Thank you so much.
Slides now. Perfect. Thank you, everyone. Sorry for that little delay. Let me start the presentation. All right, so good morning, everyone. My name is Rupal. I'm a third-year PhD student at U of T and also co-president of the U of T Stem Cell Club. I also have Kenneth CU. He's also part of the U of T Stem Cell Club. He's one of the exec member as well with our team. So today we'll be actually giving a short talk about what we do as a U of T Stem Cell Club, what stem cells are, and how we actually, why we want to encourage young generation like you and everyone else as well to educate like why we need to recruit donors, stem cell donors, to be more specific, and how the stem cells can actually change people's life suffering with different blood diseases. So without actually further ado, let's move into this. So to start with, our main mission statement is to actively engage the members of the U of T to register stem cell donors. And we also aim to use the transplantation lenses to educate students about informed consent, quality control, interpersonal collaboration, and also leadership skills in healthcare. As we know, U of T actually is really great in terms of research and healthcare. So having this club and encouraging students to actually get informed about stem cell donation is very, very crucial. So we also recruit undergraduate students. So if you want to join, please feel free to contact us after the presentation or anytime. We'll be sharing the contact information at the end of this presentation as well. So to start with, what are actually stem cells? So if you are not from science background, just a brief overview. If most of you already know, the stem cells are the cells that we actually our body uses to make different types of stem cells, as you see in the picture on the right here. So they basically they are very first type of cells when you are like tiny little seed in your mommy's belly. Those are the totipotent stem cells, and those differentiate into different type of cells to make different organs, your brain, your heart, lung, and you get the point. And this actually go into more specific type of stem cells, and those are called multipotent stem cells. So for blood, there is also hematopoietic stem cells, which differentiates into different types of blood stem cells. So we actually focus on these hematopoietic stem cells to treat different diseases for patients suffering from cancer or sickle cell anemia. Even AIDS actually has been cured using after blood stem cell transplants. So overall, it is actually very promising results have been shown after blood stem cell transplant. As I mentioned, not only cancer, but many different other diseases have been cured. And they actually do stem cell transplants from the same person themselves. So when they are actually not showing the symptoms of cancer, so when they are under remission, they collect stem cells and they store those stem cells, and when patient actually relapse, so they actually have cancer again, they transplant those healthy stem cells into the patient, and that actually gives them more life expectancy after the stem cell transplant. But not as this works for all the patients, so they have to rely on other people's stem cells which match. So they do HLA matching, and as you see in the graph on the right, that siblings are actually better match, but unfortunately only less than 25% of the time your siblings are going to be a match, so they have to rely on unrelated donors. And that's why we want to make sure that we encourage all the people from different ethnic background to register as stem cells donors, and we cannot actually go forward without leaving anyone behind. As you see in this graph, we have a very good representation of white European people on the stem cell registry, but other ethnic background people are not actually represented very well. And we really want to educate these people to make sure that they know that if they donate stem cells, they won't be losing anything. But they'll be helping people very much, and the life expectancy increases actually by 5 to 10 years, which is huge deal when you know someone is going to die in like maybe 2 or 3 months, and if you give them 5 extra years, that's actually really really an altruistic act, and you feel good as well that you actually saved someone's life by donating stem cells. So to actually fill this gap, we are educating people, running different stem cell drives to recruit people, young people between age of 70 into 35 years. So they can register, and if they are matched, they can donate stem cells and have help these patients live longer. And so overall, it's not only U of T, there are 26 campuses across Canada. So McMaster, Queen's University, Western University, all the universities, colleges also have stem cell clubs. And collectively, overall, since 2011, we have recruited 17,500 donors, which is actually still long way to go because it's not enough. And we have of this total 17,500, 58% are male, and of those 80% are between age 17 to 25. So we are actually focusing on very young. As you know, when you are young, the stem cells are also very potent, so they can differentiate into blood stem cells very easily and very nicely, and there is also less chance of graft versus host disease. So the patient who is getting the stem cells don't actually develop other type of immune reaction after the stem cell transplant. That's why the young stem cells from young population is very important to get. And also the 60% of this male actually self-identify as non-Caucasian. So as you saw in the previous plot, since we don't have as much representation of non-Caucasian population, we are actually now focusing on trying to get non-Caucasian uh, males, especially compared to females, because it has been shown by science that stem cells from males are actually more uh, reliable in the recipient compared to female. So yes, you see from this data that we have been approaching this goal of recruiting stem cell donors, which benefit the patients. And so far, we have registered like 20% of the stem cell donors recruiting to Canada Stem Cell Donor Registry every year, which is still very less because there are many patients still waiting for stem cell donors. And unfortunately, many of them, they don't find donors because finding HLA, perfect HLA match is very hard. Even 50% match is very hard. So that's why our goal is to get as many people as possible on the stem cell donor registry. So as a U of T stem cell club at St. George campus, you see we have tried our best to get a non-Caucasian male, and these are just the data from 2018-2019 and 2019-2020. As you know, we hit the pandemic, so we have tried our best to actually run many virtual drives as well during the pandemic. And these are just some of the examples of the different drives that we ran. So not only different drives like as a general drive, for example, challenges of stem cell donation, where we taught students what are the challenges and how we can actually help overcome these different challenges. And the hope stems from our donors was also a separate drive that we held. And ABC panel, we actually invited different transplant physicians and also stem cell donor and recipients. So they talked about their experiences, and also if you donate stem cell, is there going to be any complications, this and that, like all different types of questions were answered. So if you actually want to engage with us, participate in all events, you all are more than welcome to join us during these events to get more information about stem cell donation. We also actually ran very different campaigns to target specific population, as we saw that different non-Caucasian population is not represented. So we are trying to focus on those people, talk to the people within their community and find out what are the stigma and what is the hesitancy within that community, which is stopping them from registering as stem cell donors. So by actually focusing on those main points, we have shown that we are actually overcoming those stigmas and hesitancy, and people are now actually more aware, and they're registering. We're happy to actually help their children register as stem cell donor as well. So these are some of the online drives that we ran during the pandemic. And so just to give you guys overview of how the stem cell transplant works, and how you can help people by donating stem cells, we're going to show you a very short video. Let me know if you cannot hear the voice.
For many patients with blood diseases, stem cell transplantation offers hope and a chance of cure. Stem cells are factories that sit in the bone marrow and produce the different parts of our blood. Sometimes, patients have diseases that damage those factories, leading to problems. These patients may need a stem cell transplant to get better. When a patient receives a stem cell transplant, the donor's stem cells become new factories and begin producing all the blood cells needed to live. The problem is, patients in need of a transplant can't receive stem cells from just anybody. Everyone's stem cells have a specific label. Think of the various barcode labels at the supermarket. Each type of fruit has a unique barcode label that allows the computer to recognize what kind of fruit it is. Patients in need of a stem cell transplant need to receive stem cells that have the same barcode label as their own. But because there are millions of varieties of labels, the odds of finding a match are low. Most patients are unable to find the stem cells they need from within their own family. These patients rely on others to register as stem cell donors. If you're between the ages of 17 and 35, you can sign up to become that potential donor, either online or at a stem cell drive. To register as a donor, you'll need to complete a health questionnaire. Next, you'll swab your cheek. The swabs will be packed up and sent to the lab to find out if your cells are a match to a patient in need. There are two ways you could be asked to donate your stem cells, if you happen to be a life-saving match. Most of the time, stem cells can be collected from blood. The donor is first given a medication that increases the number of stem cells in their blood. Then, blood is collected from the donor, run through a sorting machine to filter out some stem cells, and then the rest of the blood is returned to the donor. The stem cells are then used to help the patient in need. The other, less common way that stem cells are collected is directly from bone marrow. Here, the donor is given anesthetic to make them sleep. and then stem cells are collected from the bone marrow in their hip. Each year, over 1,000 Canadians are looking for their life-saving match, and they're most likely to find it from within their own ethnic group. Currently, diversity on the donor registry is lacking, so there's a special need for people from all ethnic groups to sign up. Studies have also shown that treatment is most effective when the stem cells come from young, healthy males. For this reason, we are especially looking for young men and ethnically diverse individuals to get swabbed. Registering as a potential stem cell donor might take 15 minutes out of your schedule, but it could mean a lifetime to a patient in need.
So as you see, it's actually very easy to donate stem cells. You just give the stem cell, the extra stem cells that come from your bone marrow, and you get your blood back. So without actually, and your stem cell actually comes back, even the ones that you lost in only 1 or 2 weeks. So it's gain for everyone. So if you actually want to participate, please shoot us an email. And now Kenneth is gonna go over what are the steps that you can actually take if you want to register a stem cell donor today or anytime in future. Sorry.
For many patients,
Hey everyone, so to circumvent the COVID restriction problem, we've tried to recruit people to join the registry by applying online through our virtual drives. And by joining the registry online, the Canadian Blood Services will send you a package with a swab kit and the necessary information to complete the swabbing yourself. So for those that are actually interested in joining, you can start at blood.ca. And at the top, there will be several tabs. You click the stem cell tab, and then there will be a stem cell donation eligibility and registration option. And then you'll reach this page that you're seeing on the screen, and you can just click the start questionnaire button and then move on to the second step. Yeah, so in order to proceed to the next step, you'll have to do you have to accept the terms and conditions, which is that you are of age to donate stem cells. The age to donate is 17 to 35, and that you understand what entails in a stem cell to join the registry, and that you're healthy. So you just click the accept and register button and move on to step 3. And so then they will do a stem cell knowledge test, which just makes sure that you know what is entailed again in the registry, who can donate, about things about confidentiality and about autonomy. So things like if you want to withdraw at any time, you can. And then we can move on to step 4. And that's just to complete the stem cell registry profile. They'll get you to create an account where they'll collect some information, such as first name, last name, date of birth, and things like sex. Yeah, so once you do all this, it will say registration completed. And then congratulations, you're on your way to getting the kit from the Canadian Blood Services. And then you can just swab it, mail it back, and you are in the registry. So if you want to join us in saving lives, please feel free to come to a stem cell drive or joining our club. And if you're interested in that, you can follow us on our social media. We're on Instagram and Facebook, where we often post when we'll be holding our drives. You can email us any questions you have. We reply quite promptly. And lastly, if you want to join as a volunteer, you can fill out the Google form that's on this page. So I think yeah, that's perfect. So thank you for listening to our presentation. If you have any questions, please let us know.
Thank you, everyone.
Um,
I'll be also dropping the link to the volunteer form in the chat. So anyone who wants to register as volunteer, email us anytime. And we are happy to take any questions if you have any.
Yeah, so let me take out some of the questions that we do have. Thank you both so much for such a wonderful presentation. It was amazing getting to learn a little bit about how stem cells that anybody can donate have such an incredible impact on so many patients around the world. So I think we do have a few questions that I can kind of facilitate, and you guys can take your time and answer them. We have one specifically for your club. So someone is asking, what does it mean for someone to be a volunteer? What do they do? And what are sort of the different aspects of it that they'll be able to have access to?
Yeah, so that's a good question. So as volunteer, you basically help out with any activity that you are actually excited about. So the stuff that we do is basically, if you are into designing posters, you can join the promotion team and you can help out in designing different posters for the different events, different drives. You can also help out with holding the stem cell drive itself. So when you make swap kits for all the people who are interested in registering as stem cell donor. And for the drive itself, it's not in person still, because as you know, we still have the cases rising. But for the virtual drive, you can actually also participate in presenting. We won't give you all full the we won't give you full presentation to present in the beginning, but you can start with like presenting 2 or 3 slides just to make you feel comfortable and get to know what the club does, and also feel comfortable with the material itself and answering the questions. And so that's for the virtual drive. But when we start in person, which will be actually starting very soon, we are hoping from September to start in-person drives. By that time, hopefully, I will have permission from U of T, and the cases will be low as well. So during that time, what you do is, as I said, you make the swap kits for people who are interested in registering. You make sure the questionnaire they fill out is actually accurate. They filled out every single box on the questionnaire, and those are some of the very basic tasks that you do as volunteer. And as you learn more and more by participating in the drive, if you want to move on to next step and actually stay as an executive member with the club, you can do that as well.
All right, that sounds good. So as a follow-up to that, if you can explain, because our attendees are high school students, what the process of kind of signing up will be. If you want to go back, I know there was like a contact slide that you guys had. Maybe if you want to explain quickly.
Yeah, so I will actually post the link to register as volunteer in the chat, and just fill out that form, and yeah, you are good. And after that, we will send you email with all the details with the training that you need to do. It's don't get scared by the word training. It's just a very simple presentation that you just have to see, and there is no quiz or anything. You just have to make sure you understand the material. Let us know that you have done so. And besides that, we will also hold training sections where we go through the same presentation again and answer any questions volunteers may have, to make sure that they understand the process of autonomy, how to sign up people as a stem cell donor, and all the other basic questions. And yeah, actually that's it. Yeah, you just need to fill out the form for now to start with, to join us.
Okay, wonderful. Thank you so much. So we do have a, oh, sorry, were you going to say something else? All right, perfect. Our next question would be, how many times can you donate stem cells as a donor?
Stem cell donation is very specific. So you have to be actually matched to the recipient. So actually almost 100% of the time it's only once, and that too only if you are a match. If you're not matched, then you may not even get a call to donate stem cells, and you would stay on the registry until you are 65 years old. And after that, you actually wouldn't be even asked to donate stem cells, because as you grow old, you know you need those stem cells for yourself to recover and repair your body. So yeah, it's very, very rare. Only 0.01% people get matched and get an opportunity to donate stem cells. So yeah, it is very rare, but we want to make sure that people don't even lose that 0.01% chance of helping someone and live longer. So that's why we want to make sure every single person is on the stem cell registry and help those patients.
Perfect. That's good to know. All right. And then we have another question about stem cells as well. So, can you make stem cells from laboratories?
Yeah, so those stem cells are actually mostly used for research purposes for now. There are also some trials going on, as you hear, but those are not as effective. So these stem cells, if you're referring to actually generating blood stem cells in the laboratory, it is being done, but it's not in the clinical trial yet. Because as stem cells, if you don't have specific stem cells, they can actually differentiate into different types of stem cells, and it may actually cause more problem than it would help. Just to give you a very simple example, if you take stem cells from your adipose tissue, so from your fat, and if you transplant those and expect those stem cells to help you recover from your blood cancer, it wouldn't actually work. Those stem cells have already decided that they're gonna differentiate into adipose tissue. So they won't work for blood cancer. Though they actually have showed that they don't work for treating arthritis either. So it's very, very specific. The ones that they're used in the laboratory are mostly from embryonic stem cells, which are even more pluripotent. So they can not only differentiate into blood, bone, but many different types of tissues. So transplanting those into poor patient is very, very risky, and it's not actually being done as of yet. Mostly they use blood stem cells to help patients. So blood to blood is actually very safe and easy to do, and is working very nicely, but not other stem cells, and not from the lab for sure. Hope that answers your question. And yeah, Kenneth, if you wanted to actually add anything to this, you have more experience in immunology. So.
Yeah, no, I think you covered it pretty well.
Yeah, no, that sounds good. Thank you. I think maybe we'll just have one more question at the end. I know someone was asking and they were very interested about getting into the donor program. And so is there like an age, a minimum age that somebody has to be in order to go donate? And how would the process go?
Yeah, maybe Kenneth, do you want to take this?
Yeah, sure. Yeah, so there is an age restriction. It's between 17 and 35. However, when you do register for the registry, they will keep your profile there until the age of 65. But when it comes to swabbing, it is restricted to 17 to 35, just because they're looking for young, healthy individuals, which lead to better outcomes.
Okay, sounds good. I believe those are all. Oh, all right. We might have, I don't think we have enough time to go through the last one, unfortunately, but our lovely Zoom production lead did create a way of making a QR code for your registration link. So we're going to screen share that quickly. And if anybody is interested for volunteering, that will be the access to get onto the registration site. I'm assuming there will be contact information on your registration link as well. So if any of you attendees have additional questions for Rupal and Kenneth, please feel free to do a quick email to them. And I'm sure over email, they'll be able to give you a very nice detailed answer and answer any additional questions as well.
Sure.
All right. Okay, well, thank you both so much. That was wonderful getting to know that we can do this different, there's so many different things that we can do to, even if the chances are so low, to really go on and help somebody who is in need. So that's incredible work you guys are doing. And thank you all for joining us today again.
Thank you, everyone.
Thank you, everyone. Yeah, feel free to email us anytime.
Amazing, thank you. All right, so up next for our third presentation of today, we have First Robotics Canada, who will be speaking to us about their activity of Fun with SimBots. So I do also want to remind everyone again that we do have the Q&A section at the very bottom. So if you do have any questions for our speakers, make sure to put them in there. These are all super exciting presentations everywhere. So definitely throw all those questions in, and whenever we have time, we'll be having our Q&A session at the very end. So without further ado, let me give the floor to First Robotics Canada.
Well, thank you very much, and we'll just get the presentation up here and ready to go. So again, thanks to the University of Toronto Science Rendezvous programming team for allowing us to be here today. Welcome to First Canada's Coding with FTC Sim. We've called it a virtual robotics adventure, and I'm sure you'll have lots of fun with this. Fun is one of FIRST's core values, so it's important that whatever we do and whatever you do when you're involved in FIRST involves some fun. At the bottom of the screen, there is a URL that says ftcsim.org that takes you to the page for our FTC Sim interactive website that Muskan, one of our presenters, will be taking us through today. And so let me introduce myself. I'm Phil Homersky. I'm what they call a FIRST senior mentor with FIRST, and basically FIRST senior mentor helps promote the programs, and we've got programs from kindergarten to grade 12 age students as well. Help promote those programs across Ontario, and support the teams, support the programs, and basically really help get the First Canada programs going across the province. And one of our other helpers today is Aman. And Aman, if you want to introduce yourself.
Yeah, sure. So I am a recent graduate from the FIRST program. I've been doing competitive robotics for quite a while now. And I've been teaching FTC Sim, which is what we're going to be doing today, for like the past 2 years. So really love teaching FTC Sim. I think it's an amazing platform for students to be able to learn computer science fundamental principles very easily. And Muskan, if you want to take it away.
Absolutely. Hey, so my name is Muskan, as mentioned. Thank you for the introduction. I've also been in competitive robotics for around 5 years now. Learning and STEM, it's a big passion of mine. So today, what I want to do is get you guys to jump into looking at how robots work, and specifically how they're programmed to do the incredible things that they can do.
That's great. Thank you very much, both Aman and Muskan. So before we get into what we're going to do today, I just wanted to go through what FIRST is. FIRST is actually an acronym, and it stands for Inspiration and Recognition of Science and Technology. And the mission of FIRST is to inspire young people to be science and technology leaders. and innovators. And how do we do that? Well, we do that by, as I'd mentioned before, we've got programs from JK to 12, and so we do that by engaging young people in exciting mentor-based programs that build science, engineering, and technology skills and inspire innovation. And we have programs for students as young as 6 and right through to high school age, as I said. And for more information about our programs, feel free to visit the First Canada website at firstroboticscanada.org. We'll put that on at the end of the presentation. Today, we're going to be focusing on the First Tech Challenge there, that middle one, and that's why I'm wearing orange, because we've got colors for each one of our programs, and orange is really the First Tech Challenge color. So we'll be profiling some of the work that you would be doing, coding and making robots move through the First Tech Challenge SIM programming. So without further ado, I'm going to pass it over to Muskan, and then she can take us through the FTC SIM virtual robotics adventure.
Perfect. Thank you so much. All right. So I would like to just firstly make sure that everyone is on FTC SIM. So if they would like to follow along, so I'll give you about 10 seconds to get on there if you aren't there already.
So I think it's gonna take a little bit more than 10 seconds because I'm not sure if they've all received the link, the actual instructions to log in. So maybe I think what you should do is you should log out and then show them the process of creating an account.
So if we go to ftcsim.org/ftcsim, which is the link that you were all given, it should take you to this screen. And I believe I'm probably logged in right now. I am. Hold on. Sorry about that. So if I log in, okay, sorry about the credentials on there, but you should get a username and password input, which should get you logged in. If you don't have one, I don't believe you should need it for now, but you should be taken to this screen. And I would like you to press FTC movement, this one over here. So following along, when you click there, click the first one, line. I'll give you some time to get to where we are right now, but that should bring you to this screen. Don't worry about the video, we can talk about everything together, but you should come to this page where you'll see a little robot and a flag at the end. So hopefully you guys are at this area. So just to get started, can someone tell me their idea of what a robot is? No wrong answers. It's anything. How do you define a robot?
And so I'm just wondering, for the organizers at the Science Rendezvous here, what permissions do the participants have? Are they able to type in chat, or are they able to turn on their mics?
Yeah, I was just gonna say that. So they won't be able to put in the chat, and they won't be able to put on their mics just for privacy reasons. So we can, they could type into the Q&A, and maybe I can kind of incorporate their answers to the discussion if that works.
Sure, yeah, sure.
All right, okay, so a participant says that robot is a machine created to complete a task, usually with a computer system in it.
Perfect, yeah, I love that answer. Robots are used to help progress humanity work for a task, and we're getting constantly more intelligent with them. So what you guys see on screen here right now, this little guy over here, I hope you can see my cursor moving around, that is a little virtual simulation of a robot. And what we're going to try to do here is program this robot using these blocks. I can walk you through them in a second, to get the robot to move forward and get to this flag. So the first thing that we want to do is obviously look through our code a little bit. So it's here in FTC SIM, it's been divided into different sections. You can find different blocks that will do certain things. And if you there's actually going to be a list of the total amount of syntaxes or different blocks that you can use if Armand would like to throw that link into the chat with the full list of them. But just to get started, so one thing that we need to do with robots, which is a little bit counterintuitive. Actually, why don't I open the floor? What would you do? How would you get the robot to reach the flag? Anyone can answer. what do you think the first step to get a robot to move would be? Think of it like a car. How do cars move?
Yeah, so we have an answer here. So we need to have a motor and get the engine to start.
Perfect, yeah. So on this robot specifically, we don't have something exactly like an engine, but what we do have is motors, which you can see right here. So the thing with this robot is the motors are oriented in a specific way. So what we're going to do here is we're going to reverse one of these motors. And what I just want to show you really quickly, I'll do a little bit of an explanation in a second, but if you look at this code, I know it can look a little overwhelming right now, but if you see here, run op mode simulator, and right under it says put initialization blocks here, what that means is that's the first section. This will run all this code immediately as the program starts to run. When the simulator is running, it's going to do everything underneath the initialization blocks. Everything under here, call simulator, wait for start, and then the repeat block. What this is going to do is it's going to be doing all that code underneath there while the code is running at all times. So it's always looking to do any code that's in here. Currently, it's empty because we don't really need to do anything like that, but it's to keep in mind. So back to motors. If we take a look at these motors, it's very difficult to see on the actual screen, but the orientation of the motors are, let's say the motor was like this, it's going to spin this way. But if it was oriented upwards, in this case, which it is, when you spin the same way, it's going, when you spin it in like clockwise, for example, it is going to go counterclockwise. So just keep that in mind for now. So what we're going to do here is we're going to set the direction of the left motor to reverse. That way, when it's oriented in this manner, both motors are going to go in the same direction since they are oriented differently. So then what we wanted to do, as you guys mentioned, is we want to make the motors go, right? We want to get it to go touch the flag. So if we go to DC motor, I hope all of you guys are able to follow along. Just to save some time, you could take it from here, but if you click dual, you'll get a set power motor left and motor right block. So if we put that right under here, because remember, we don't want to put it in the run blocks because that's going to be doing it forever. And then we're never going to be able to stop it, right? Because it's going to be constantly looking for it. So in the initialization, we set the power to the motor. So here we can see that we're setting power to both the left and right motors since it is dual. So let's check it out. Let's see what happens. Okay, we got somewhere. So we didn't pass the challenge just yet. And that's because we didn't, we went past the flag, right? We didn't stop at the flag. Can anyone try to tell me why that happened?
I have a question for you, Muskan. So what would happen if we didn't reverse the direction of the motor, left motor? Could we see what would happen if we didn't do that?
Absolutely. Let's take a look at it. So I'm going to disable this block for now. So if you right click or double click on any of these blocks, you'll get this pop-up menu where you can do some fun things with it. Here, if you click disable block, it won't delete it, but it will keep it there so that you can use it whenever you like and you can enable it back again later. So let's put in the same blocks again one more time. This time, oh, sorry, I did the wrong one. I didn't reverse the motor. My bad. So let's disable this block. And then we can put this back in here. I will enable that block once more. So let's take a look and see if we just power the motors forward without reversing the direction of the motor. Let's see what happens. So as you can see, the robot is constantly spinning. And since the orientation of the motors are different, they're going to be opposing each other, not really moving in the same way. So we fix this by setting the orientation of both motors the same in the programming. So we make the program consider each motor as the same by reversing the direction. So let's enable that again. And did we get any answers on why we think that the robot didn't touch the flag? No answers? All right, that's fine. So if we look at it this way, let's look at actually what we have told the program to do. So going through, we got it to reverse the direction of the motors. That way it can go in one straight line since the motors are not opposing each other. And then we got the motor power set so that it would move forward. But what we never told it to do was to stop the motors. So why don't we try that again? Let's go back to dual. Let's get our motors and let's put it here. So if if you can see, set power motor left 1 and motor right 1. 1 is the highest speed that you can get to with the motors. So you're going to move it as far as you can. -1 would be reversing it, going backwards, and 0 would be no power at all. And that's what we want to do here, right? We want the robot to stop. So let's set the powers to each motor to 0 and see what happens. So if you look at that, nothing actually happened with the robot, and you might be wondering why. So let's take a look at it. What you need to know about robots and computers in general is that they take instructions very literally. They don't consider the actual objectives and goals of what they're trying to achieve. We know that the goal is to make it to the end of the flag, but what it doesn't know is that it also has to do that. So it can't really conceptualize that it needs to go for a while until it touches the flag and then stop. So what it's going to do is it sets the power to the motor, even though you couldn't see that. It looks like it set the power for left and right, but since computers think so fast, it only went for such a short amount of time that you weren't actually able to witness it move, because immediately after, we set the power back to 0. So what's the solution here? So one thing that we can do is we can tell the computer that, okay, let's set the power forward so that it can move, but only for a certain time. Let's ignore everything else we have in the code, and for this much time, let's only move forward. After that much time has passed, then let's continue reading. So we can do this if you click linear op mode here. You see this block, call simulator sleep, milliseconds 1000. So if we put that between the setting the motor power forward and stopping the motor power, what this is going to do is it's going to check the simulator after it's set the power, and it's going to sleep for 1000 milliseconds or 1 second, which means that it's not going to read any of the code that's underneath that, unless it's in the run blocks, but we don't have anything there, so no worries. But for now, it's going to stop checking anything under the initialization, including the stopping the motor power. This means that the robot should run for 1 second or 1000 milliseconds, and then it should set motor power to 0. So let's check out what that does. Okay, we've made some improvements. The robot successfully was able to move forward, and it was able to stop. Our problem here is that it didn't actually reach the flags, correct? So what we can do is, if we wanted to run a little bit longer until it gets to the flags, what we should do is we should make it run, we should make it move the power longer than we have for 1 second. So if we increase the value here, let's say, where did it get, about here? So let's try 1100 milliseconds. So at this point, what it's going to do is it's going to keep going, and it's going to go a little bit farther than last time, because it's going to stop reading to set the motor power to 0 later. So it's going to keep on going for an extended period of time until after 1100 milliseconds passes, it's going to stop. So here we're going to have to try to find the value in which the actual robot is going to hit the flag and then stop. Let's take a look.
So if we can take a pause here, and hopefully people can try to catch up to this point, and maybe they can try testing out their own values for the time, and then you can put into the Q&A section what values you found that works for you. So we saw that 1000 was a little bit too less, even 1100 was a little bit less. So try experimenting with your own values and see what you get, and let us know what value you find that's able to touch the flag, complete the level, but not go past it and crash into the wall. And I did want to point out something interesting that I saw in the Q&A. Someone said that you could use an if statement to check if it's touching the flag and then stop based on that. So that's certainly true. It's quite an interesting thing you can do. And in fact, it's a more advanced method of coding. Because in the real world, time-based code is not always that accurate, since you could have your wheels slipping on the ground, or your encoders could not calculate the total revolutions correctly. So by checking whether or not you're hitting the flag, say maybe through the use of a color sensor or a distance sensor, that'll always be more accurate. And in fact, that's a better way to code. And what we really encourage you to do is, if you enjoyed the session, we want you to go look at other levels in the FTC sim, because there are levels that you can take advantage of using the color sensor and distance sensor to check if you're at a certain spot. And at those times, that's more advanced coding, but it's certainly really, really cool. And we really encourage you to go try that out.
Absolutely. So I think we do see in the Q&A, someone wants to try 1500 milliseconds. So let's try that in. If you guys would like to explore FTC Sim after the session, we highly encourage you to find different and creative ways to achieve the same goal with different lines of code. But for now, let's put in 1500 milliseconds and see where that takes us. Perfect. So as you see, 1500 milliseconds here was enough for the robot to touch the flag without going far over it and touching the wall, which means that we completed the puzzle. So what I want to do here is just go over the code very quickly so we can go make sure that everyone understands what's happened. So what we started, we reversed the direction of the motor. So since it's oriented differently, when it's reversed, it's going to go in the same way as the right motor. Then we set the power to each motor to ensure that they were moving forward, because 1 is the maximum speed of moving forward, -1 would be backwards, and 0 would be stopping. So after that time, we experimented a little bit, and we found that the simulator should run for about 1.5 seconds, or 1500 milliseconds here, before it needs to stop, because that's how long it would take to get to the flag. After that time has elapsed and it's reached to that point of the flag, we set the motor power to 0. So once you set it to 0, the motors are going to stop moving and it's going to stop at the flag. Now, what I want to try is if we change the motor power 1 to 0.5, what that's going to do is it's going to change the speed at which the motor is running. So now it's going to move a little bit slower. So let's see what happens again, just with no other changes. So we see that it's moving slower, of course, and it hasn't made it quite to the flag yet. Does anyone want to tell me why, or does anyone have a solution that they can come up with?
So we're going half as fast, correct, Muskan?
Yep, we are going half as fast. So what is one way that we can get the same result in a slower medium?
We're going half as fast, but we're going for the same amount of time, right? We haven't changed the time we went.
Yep, correct.
Okay. So let's see if anyone has some ideas. And let's say we can't change the time back to 1. We want to still go at 0.5. So the only variable here that we can change would be the time we drive for. So if anyone has any ideas whether we should increase it, decrease it, and by how much, we'd love to hear them.
3000 milliseconds, says Sahil. So let's try that. That makes sense. It is double. We decreased our speed by 50%, so we might want to increase our time by 50%. Let's take a look at that. Perfect. See, so when we do programming, there are many different ways that we can achieve the same result through different types of code. And here we see that because we don't always want to be rushing right to the end. We don't want to be using full motor power at all times in the real world. In a simulation, it can work out because it is all virtual, but when you apply the physics of real life into the world, if it's going too fast, it may harm your robot. You may miss something. Your sensor might zip by too fast to notice something. So we want sometimes to go at an appropriate speed, which means that we may have to go for a little bit longer. And as we had mentioned slightly before, it is important to think of ways to code so that isn't so rigid, like moving by time. So it would be better, of course, to be able to detect the flag and stop until you see it. But in this case, we found another way that we can make an accommodation if we only want to go at 50% speed to still get the same result. So we are nearing on time, but I do want to check out the other puzzle. So if you all click yes on puzzle complete, I think we can get started on here. And I'll give you a second to get there. Oh, my code is here. That's not fantastic. Okay, that's unfortunate. I will remove them. But I hope in this time you guys can all get to the second puzzle.
I think no, we still want to leave some time for questions with people. So I'd say we just kind of get them to think about how they would solve this one. And then, we leave it at that because I don't think we have enough time to go through the full thing.
Sure. So here the goal is, if you can see the robot, it is actually oriented backwards. And you could say that, okay, of course, we can just set as I mentioned before, we could set the motor power to -1, which is it's a much easier solution to this problem. But if you would like to start to get turning, can anyone drop an idea of how they would turn the robot to make it front facing so that these wheels over here would be facing the flag to get it to turn around and move forward? We already know how to move the robot forward, but how would we get it to turn around and get there? Do we have any ideas?
And what I like to point out is it's funny because we've already done that.
We have.
Before we reverse the motor, that's why I asked what would happen if we didn't reverse the motor. We noticed that the robot was spinning. So how do you guys think we can get the robot to spin this time intentionally, though?
So Sahil says have a difference in motor strength. So do you mean as in for left and right? So we would increase the power to the motors, which we did try before. The maximum speed for these motors is 1. So we would doing something like 2 wouldn't be the most helpful here. But if we think about it this way, how can we use the motors individually to get it to spin around? Because if you think of this robot, you can divide it into 2 halves. The left half controls the left half of the robot, and the right motor controls the right half of the motor. So if we increase the power of the motors, it may go a little bit faster, but I don't think it would turn around.
Well, I think
Left with 0.5.
Yeah, it's quite interesting because what he's describing there is something we call a point turn rather than a swing turn. So what you're describing there, Sahil, is turning along a radius. That would mean kind of following the circumference of a circle with a given radii and making a turn that way, which is very interesting. And in fact, if you look at the level right after that, that is actually the optimal solution to solve the maze, is using something like that, a point turn, which functions by having a difference in speed between the 2 motors. However, what we want to do in this case is something called a point turn, which instead means instead of turning on a radius, we turn right on the center point of the robot. We turn about that point. So to do that, what you actually want is to have your motor powers not just different, but in fact have the same magnitude, but in opposite direction. So if you have one set to power 1, you want the other one set to power -1. Or if one is 0.5, you want the other one to be -0.5. Having the same magnitude but opposite directions allows it to be equal, and it would actually turn on the spot rather than make a swing turn. And the reason we don't want to make a swing turn is because if you look at the walls, they're very close to the robot, which doesn't leave enough space for you to make that long arch turn out. You really want to make a tight turn right on the spot. But I think that was an excellent guess. And certainly if you had a little bit more space in that maze, your technique of setting the power to 0.5 and 1 would definitely have worked, and you would have made like a long turn and swung back around for sure.
So very quickly, let's take a look. So what I've done here is I've removed the reversing motor block. So now let's just set the powers of the motors to 1. And we did this before. And if you take a look, it does start to spin since the motors are oriented in different directions. So what we would do here is we would get it to stop after it's made a 180-degree turn, because that is where we want it to be oriented. And then we would make it move forwards once again. So in this case, I will not go through the full extent of the challenge, but I do highly encourage you to all try this challenge on your own after this session is over. So you would get the robot to spin and make it stop using the sleep block as we tried before, and get it oriented exactly to where you would want it to be. And I will leave the rest for you to explore. And why don't we now open up the floor for any questions about robots, programming, or how we got any of this done.
Muskan, we do have a little bit more time. I unfortunately, I thought we were going to 11:50, but we've got until noon. So
okay.
If there is a possibility of moving through another session that might be of interest to the participants, since we don't seem to have any questions at this point, maybe we can just move on to another demonstration, if that would be okay.
Perfect. Okay. So what we can do here, I'll quickly show this to you because I find there's something actually more interesting I would like to show you. Actually, I can teach you that later. Why don't I take you guys? So can you all go back to ftcsim.org/ftcsim again, the initial link that was sent in the chat, and go back to FTC movement? I'll give you a second to do that, but what I want you to do is click number 6 square here, because that is a very interesting one that I would like to show you all. So I hope you can find your way to challenge number 6 square. And this can be a little bit daunting, but just using what we remember. So as you can see here, the flag is now in a little bit of a weirder position, and your robot is facing the wrong way. So as I had mentioned earlier, we do have something here called run blocks. And run blocks, as I had said, they do a loop constantly. They're checking for something. So we can take an advantage of this loop. This loop says repeat while the call simulator op mode is active. So while the simulation is running, it's going to constantly do these things. But if we go to loops, we can see that there's a repeat block for a certain amount of time. So let's check out what we can do with this. When we think about coding, our goal is to be as effective as we want to. So what we could do, let's follow our path here. Let's look at the route. So our robot is currently facing the wrong direction. It would move forward and hit into that wall. But as we learned before, we can spin the robot around by altering the motors, and we can get it to do a turn, in this case about 90 degrees. So the robot won't actually know what 90 degrees is. We will have to find the time. But once it's facing the correct orientation on the field, we would want it to go forward until it gets roughly close to that edge over there. And we would want it to turn right again to go to this area. Go forward, turn right, go forward, turn right again, and move forward. So as you can see, there's a lot of repetition in here. And I hope we can all agree that it'd be pretty annoying to write that same piece of code over and over again. So let me show you something cool. Let's count the amount of times we have to do the same thing. So here we want to turn right, move forward. That's one time. Turn right, move forward the second time. So then you would be in this area. Turn right, move forward one more time. So now you would make it here. And turn right, move forward another time to get to the flag. So that's a total of 4. So if we do repeat 4 times, let's try this. So everyone remembers, if we set the motor direction to reverse, it's going to go straight. But right now we want it to turn right. So why don't I just disable that block for now, because we will need it later. But let's take a look at this. So if we go to dual, let's set both powers to 1. And let's get it to stop after, say, 500 milliseconds. Why not? Let's see where that takes us. Oh, I forgot to stop it. But okay, so we all remember that after the sleep block is placed, we would have to turn off the motor power. So let's see where 500 takes us. So that seems like definitely way too much, but we remember that is actually repeated 4 times. So sorry, okay. I messed up a little bit there. We would have to go a little bit farther. So we first of all noticed that sorry, continue.
No, I was gonna say, so we do have a stop at 12, and and we'll want to have some time for questions. And I think I have a model of this maze done, so maybe I'll show that off really quickly. So
absolutely, go ahead.
Yeah, so if we quickly look at this, because it takes some time to kind of get all the numbers tuned in, and sometimes seeing the numbers takes quite a bit longer than we would like. But in this case, we can see that it's the exact same logic that Muskan was trying to explain here. We're going to repeat 4 times, and what we're going to do within that repetition is we're going to make a turn for a specified amount of time, and then we're going to drive straight before stopping. And we're going to repeat that 4 times to basically go around the square. So if I run that really quickly, you'll be able to see that all take place. We're going forward and returning, and that would have been 1 cycle. And now we're going to do that 4 more times until we reach the flag. So again, you'll see that last one, it goes kind of crashes into the wall just because all the other side lengths had been longer. So, it's not ideal, but it does work really efficiently to get the maze completed. And so I think that's pretty cool. One important thing you can remember is that when you're doing a loop, kind of like Muskan found out the hard way, is that once she had it spinning, but did it 4 times. So it went 4 times as much. You have to be kind of careful what you're putting in a loop to make sure that truly you are able to repeat exactly what it is that you want to repeat inside that loop. And secondly, I wanted to show off, so I was talking about that swing turn, but I was, you kind of have to imagine it in your head, and really I wanted to show it off right over here. So this was that level I'm talking about with the swing turn. And notice that it has this kind of arch shape. So I forget who at this point had made the suggestion. I think it was Sahil, who had made the suggestion to set the different powers and motors at different levels. So in this case, we're setting them to 25%. The right side is being set to 25% the power of the left side, and we're using that to make the turn. So if we actually run the block, you can notice what I'm talking about how you're turning on an arch. You're turning along the circumference of a circle, and that's what we like to call a swing turn. So you've seen a point turn, which Muskan showed off in level 2, and this is what would be a swing turn. So depending on the ratio between the power on your right and left motors, you're able to change the radius of the circle that you're essentially tracing out. And so by finicking with this one a little bit, we were able to get it to perfectly follow the shape of that archway. So that's kind of an application of what we were talking about there. And since we only have 5 minutes, I think now would be a great time to open up the floor for any questions people have about FIRST or about the coding we've done here today. In the meantime, I can, while this questions start rolling in. Okay, so I think I see one from Sahil. Would you like to take that?
Sure. So this coding language is actually developed by FIRST itself, and it is a block code. It's translatable to Java, but it is specific to the FIRST robotics competition for the robots that they code. You will find similar logic in all different kinds of platforms that you use, but the simulator is in a block form of something similar to Java.
And in fact, you are actually able to convert the blocks that you write into Java. So if you click on the upper right-hand section of the of the sim, you'll be able to see that it takes all the blocks you've written and it converts it into Java. And at that point, you're able to start natively coding in Java, which I think is really cool. It's a really cool way for more advanced programmers to take their skills to the next level.
So while other questions roll in, I would like to talk a little bit about FIRST itself. So as Phil mentioned earlier on in the session, FIRST Robotics Canada FIRST Canada is a company that is hosting robotics championships from all ages, from kindergarten into grade 12. And we participate in many different competitions where we build and program robots to do different tasks, and the challenge or the game of every year is different. This year we had a lot to do with freight, and collecting and picking up and moving freight, which you would be able to do when you program certain motors to do certain things with a unique robot you built. So what you guys were looking at is the programming language for the first tech challenge, as he had mentioned, which is the middle-ish group for the middle age range. And it's a smaller scale of very large industrial robots where you would solve the challenge, where this year there was cubes and spheres and also even ducks that you would have to pick up and maneuver around the field. So I have been in First Tech Challenge for 3 years now. And if you guys are interested, I highly, highly recommend that you check out FIRST and you go into their program and see if you would like to join a team.
Yeah, thanks for that, Muskan. I think that's important. If anybody is interested in looking into being part of the First Tech Challenge program, certainly come visit our First Robotics Canada website, and that URL is on the screen now at the bottom there. And you can also check out some of the other programs that Muskan mentioned. Most of these programs are done through schools, but we've got other community teams as well. So even if your school isn't able to participate in a First Tech Challenge or any of the other programs, then it is possible to get a group of interested friends together and put a team together. And as long as you've got a couple of adults that can help mentor the team and help you with all the components, I think you'll find it's a very fun time that you will have with that. As Muskan says, there's every year there's a new theme. It was transportation last season. This new season is about energy, and in most of the programs that we have as well, it's not just about the robot, and that's one of the one of the taglines that we always have. It's not about the robot. There's lots of other things that you can contribute to in these programs in innovative ways of dealing with the themes. So, for this next season, then the theme of energy, then how can you you look at a real life situation and you postulate how you might come up with a solution to an issue or an opportunity that might be using robots, but could be something to do with the stuff that you've learned about in developing your robot as well. So there's lots of fun stuff that you can do with that. Are there any other questions before we before we wrap up?
I don't see any, Phil, but if anyone needs any extra help with FTC SIM from a technical perspective, there's a lot of links in chat, so maybe copy those over before this webinar closes down. So just copy them onto your own system. There's video tutorials, there's documentation, there's a whole bunch of stuff. If you're interested in continuing on using FTC SIM, which we highly recommend you do.
Yeah, I think that's great. Aman, you did put some links in the chat there. I'm hoping that they can be shared with the participants as well. But just a reminder, yes, to go to the firstroboticscanada.org site. But Armand also indicated that there are a number of tutorials for the FTC SIM that we just went through as well that are contained within that URL that you can check out, and they're very instructive and helpful to get you through some of those perhaps more challenging sections of that. But I do recommend that you come back and visit the FTC SIM on a regular basis and try it out. It's all trial and error, and it's all a lot of fun, and it's all about coding, and I think you'll have fun with that.
Amazing. Thank you to the three of you for really educating us on robotics. I mean, I thought it was super interesting. I'm sure, I think there were definitely a few people in our attendees who were super interested in this as well. I think today we got a super interesting view of a whole field of sciences, and just, I guess, getting to see something as simple as telling a machine to move, the whole coding process that goes behind it. I mean, I'm floored. I've learned so much. Thank you.
Thank you so much for having us. This was a pleasure.
Thank you. Perfect. Okay, so with that, First Robotics will help us transition into our intermission break for today. So we had a lovely morning of amazing talks. And since it is noon, maybe grab something to eat, take a bit of water and have a little break. We'll be back here at 12:15. So if you guys want to take a good 15 minutes, we will be resuming the show shortly. And again, if you do want to come follow us on our social media, we do have updates as well as giveaways. And if at any point you want to go back and look at the schedule again, it's just given at the bottom of our page over here at www.sciencerendezvous.uoft.ca. Okay, and with that, we will see you all at 12:15. All right. Hi, everyone. Welcome back. I hope you got a chance to get some water, get some snacks, and we're just going to jump right in now. So first up for today after lunch, we have the Department of Molecular Genetics, who will be presenting to us the first of their presentations. And this will be specifically on participating in science through video games. And before I hand it off to them, I'm just going to remind everyone once again that if you do have any questions, please post it into the Zoom chat. And myself, Carrie, your MC for today, as well as the department will be able to take up these questions as they come, but we'll dedicate a section at the end of the presentation if there is time to kind of go over and answer all of them. All right, so if I can hand it off to our presenters from Molecular Genetics. Go ahead.
All right, hello everyone. I am going to start off, just share the screen. Okay. All right, hello, my name is Kiki, and I am a graduate student from the Department of Molecular Genetics. And today, me, Tim, and Matt, who are also fellow graduate students, we're here to show you how you can actually participate in science through playing video games. We're hopefully going to be able to show you tutorials of 2 ongoing science projects and also give you some time to actually try these projects yourself. So first, I'm going to give a brief introduction of these science video games and why they are different from our normal mainstream video games. So first, what are citizen science projects? Well, citizen science projects are actual science research projects that are led by trained scientists. So these are the scientists that you typically imagine as being stuck in a lab all day. But instead of doing wet lab experiments every single day, these projects involve public participation in data collection and analysis. So in other words, you, the public, become a part-time scientist. And many of these citizen projects typically involve some kind of ecological study. And here on the right, I'm showing you a poster of a city nature challenge that recently ended. And the City Nature Challenge asked the public to please take pictures of local wildlife and upload it to an app. And the end goal of this project is to essentially have a good surveillance of all the wildlife that is in a specific region, and in the hopes that maybe someday this data will be able to help ecological protection. So can you become a scientist by just playing video games? Well, you actually can, and I'm actually going to show you an example of one called Project Discovery. So what is Project Discovery? Project Discovery was a project that was hosted on this MMORPG game called EVE Online. And this project ran for a year between 2017 to 2018. And there were 320,000 players that were involved. And here's just an interface that's showing you what the game looked like. And the goal of the game is to essentially help scientists answer, where are these proteins? So here is an example of what you would come across in this mini game. So here on the left, we see pictures of human cells. Cells are the building blocks of human tissues and organs. And you can see that there are these green dots, also a blue region that indicates the location of what we call a nucleus that contains DNA. And there's also these red regions that are marking the outside of this it's part of a cell called the cytoplasm. And essentially in this game, you're asked to identify where is the location of these green dots. So on the right, you would be able to click these answers. And there are also example images of each of these locations. So the nucleus would mean that these green dots are within this blue region. Cytoplasm would mean that the green dots are found in the red regions, whereas periphery means that they're found just slightly outside of the red regions. So just by looking at the cell, we see that most of these green dots are found within this blue region. So you would be able to click on this answer, nucleus. So you would so what happens actually to the answers? And the outcome of this project was actually a creation of this automatic tool where essentially you can just input an image, and this tool will allow automatic identification of these protein locations. And here is an article that was published in 2018 that describes the outcome of this study. And if you're interested, I highly recommend that you look it up. So if you're interested in looking for other citizen science projects that are ongoing in Canada, you can actually go to the Government of Canada's webpage called the Citizen Science Portal. And I will upload this link into the Q&A chat. So you can all take a look at it when you have time. So now I am going to hand it off to Tim, who will actually show you our first citizen science project game that we would like to introduce, and it's called Eterna, which is an RNA folding game. So I'm going to stop sharing my screen and go to Tim.
All right. Thanks, Kiki, for the introduction. So, sorry, I can't see my presentation. Okay, try again. Okay. So I'm going to give you guys a brief introduction to nucleic acids, which are what we are commonly referred to as the building blocks of life. Some of you may be familiar with this pathway that I've diagrammed here. We call this the central dogma. That is DNA, which holds the genetic information in the cells of our body, is transcribed to RNA, which is thought of as a blueprint to make proteins, which are the molecules that drive chemical reactions in cells. And RNA is super interesting because not only is it a blueprint, but it's also actually capable of doing both the tasks of DNA and proteins. And nucleic acids are also interesting because they can fold into different shapes based on the sequence of its component bases. So bases are the building blocks that make up DNA and RNA. And what's special about bases is they are limited in how they can pair with each other. So as I go through the game, I hope you will be able to appreciate why base pairing is important. So the problem here is that it's very difficult for scientists to take a DNA or RNA sequence, like the one I've depicted on the left here, and predict the shape that it might fold into. So in Eterna, scientists develop puzzles that are based on the shapes that they'd like to create. And your role as a player is then to provide sequences to fulfill the requirements for each puzzle. And going from there, scientists can then take these sequences, go back to the lab and perform tests to see whether these sequences are appropriate for the questions that they'd like to answer. So Eterna has been really successful over the last couple of years in answering actually a lot of different scientific questions. And one of these things is that the players have helped to make new algorithms to figure out how RNA can fold. And it's also even been applied to stabilize RNA that's been used for RNA vaccines, such as those developed against COVID-19. So now what I hope to do is to spend maybe the next 10, 15 minutes to play through the tutorial, and I'd like to invite everyone to play along. You need to just go to eternagame.org Here you can simply press play now to start playing the tutorial. Or you can actually download apps to play on a mobile device. But what I hope to accomplish here is for you to understand base pairing and how RNA can fold into different shapes. So I'll just go ahead and I can share the link in the chat first if you need it. So I'll just stop sharing and switch over to the game. Okay, so this is the first puzzle that you'll find when you press play. So I'll go ahead and start it. So this is the interface that you'll see when you start playing the game. In the top left corner, you'll be able to see the requirements for each puzzle that have to be satisfied. So here it's simply asking for 4 or more blue, red, and green bases in the RNA that's presented to you. And on the bottom panel, you'll be able to see the controls to interact with the puzzle. So by clicking on each of the colored circles here, you can choose to mutate, to change each of the identities in the RNA that's presented here. So here you can see all the bases in this RNA species are yellow or A bases. What we can do is click and change the bases to blue bases or U bases here. And by doing that, all I have to do here to solve this puzzle is incorporate 4 or more of these different colors into the puzzle. So I'm simply clicking on what I would like the base to be changed into, and then clicking on the base in the RNA to change it. Okay, so as you can see, the first puzzle is really just for you to get used to changing bases. So there's no actual pairing in the first puzzle. Okay, so someone mentioned that it's their puzzle is still loading. I think that's fine. I just wanted to, because we have a limited time, I'm just going to continue to play through each of the puzzles, and then hopefully if we have some time at the end, then I can answer more questions. For the puzzles actually get quite a bit more complicated, and the tutorial has about 20 or so different puzzles, and you really do have to play through all of them to be able to start tackling some of the puzzles on the platform. So it's not something that you'll be able to start right away, but if this is something that you find interesting, you'll be able to learn a lot about RNA just by playing through these puzzles. So the next puzzle is where we start talking about base pairing. Okay, so in the top left window, you'll notice that there's an additional criteria that's been added. And this is representing what we call the secondary structure of the RNA. So that's the shape of the RNA once it's been folded. So here it's indicating that we need to make sure that it folds into actually the shape that's being displayed on the screen right now. And this is, we can actually toggle between what the RNA looks like in its natural state right now by clicking on this leaf. And if we click on the bullseye icon, we can actually see the desired shape of the RNA. So to solve this puzzle, we have to make sure that the bases on either side of this this contact surface here actually pair with each other. So you'll see here on the criteria that it's showing you that the yellow circles actually pair with the blue circles, and the blue circles can also pair with red circles, and the red circles pair with the green circles. So these are what we call AU base pairs, UG base pairs, and CG base pairs. So to solve the puzzle, we simply have to change the bases so that they start pairing. So here I've made an AU base pair. I can make a UG base pair here in the middle, and I can make another AU base pair here to satisfy the puzzle. Okay, so next puzzle is slightly more complicated. We have to take into account that it's asking for more base pairing in the locations that are indicated in the top left. Here it's introducing the two view modes that I already introduced, the target and the natural display. And okay, so to solve this puzzle, we just have to make sure that all the bases that are aligned right now actually pair with each other. So we can do that by making AU base pairs here. And if you zoom in and out with your mouse wheel, you can zoom in on a specific part of the sequence. Here I use the same strategy to make base pairs. Okay, so here another similar puzzle. It's asking you to make base pairs with these contacts here, but it's not giving you any kind of other constraints. So you're free to make AU base pairs or CG base pairs or UG base pairs, unless the bases are actually locked. So if you see there's this little key on some of the circles, the bases, that means that you can't change their identity. So you need to take what you know about base pairing to solve the puzzle. So if this is a locked A base, you know that you would need to use a blue base here to satisfy that base pair. Okay. Okay, so with that in mind, I can do the same thing. Here I see that there's a locked green base, so I know that a red base would be needed to pair with it. The same with the opposite side. And here, same thing applies. I use a G to satisfy. Okay, so as you can see, I've finished the first mini section of the tutorial. Now it's going to teach you about which base pairs are actually stronger. So this becomes important when we're trying to solve puzzles that are asking you to make different secondary structures. So here it's asking for 3 or fewer CG base pairs. So it's actually teaching you in the tutorial to consider using these base pairs at the kind of the terminus of each kind of what we call loops here. So that's what I'm going to do. I'm going to introduce CG base pairs at the bases of each of these loops. Okay, And I can have one more CG base pair, so I can put it here, for example. And then the rest I have to change. So this would be an AU base pair. This could also be an AU base pair. And then these two could also be AU base pairs. But you see that I need to incorporate one or more UG base pair to solve the puzzle. So I'll do that by changing any of the AU base pairs that I've created. Okay. Sorry, I haven't been timing myself, so I don't want to take up too much time from Matt's presentation, but do you think I should go through another puzzle, or do you want me to stop and let people?
I think you can keep going. I think we're going until about 1:15, 1:20.
1:15,
so
okay.
Maybe give them, you guys got like another 10 more minutes.
Yeah, okay. So the next puzzle is teaching you about the orientations of each base pair. So it's introducing this swap tool that's highlighted by the yellow arrow right now. That basically lets you swap the position of the bases in each base pair. Okay. So here it's actually telling you that even though all of the bases are paired in this RNA right now, it's actually not solving the puzzle. So what we have to do is play with the swap tool to see if the orientation of the bases make a difference. So actually, one thing about these AU base pairs is that they like to be oriented in an opposite manner. So they call it an AU zipper. So what we can do is just make them all opposing each other so that we can solve the puzzle. All right, here it's going to introduce a little bit more about the rules for or guidelines for solving puzzles. So basically teaching you kind of a basic strategy to solve the puzzles. You can skip ahead and start playing through. Okay, here you notice that I don't have any base pair constraints. So what I can do is start by just making the pairs. Okay, so yeah, I'll play it for 5 more minutes and then Matt can take over. Okay, so I can do that by start making base pairs. Actually, if you there's a hint window here. Actually, if you can't figure out all of the puzzles here, so here it's suggesting that it's indicating that at base 10, so this what's currently a U that's pairing with the A here, suggesting that I need a strong base pairing to help this particular part of the RNA fold. So what I can do is, sorry, trying to get the zoom window out of my way. What I can do is try to swap the direction of the base, but you see that doesn't work. So what I can also do is just change it into a CG base pair. Okay. Oh, you can see that a CG base pair works, but only in one particular orientation. Okay. So the next puzzle is teaching you how to use the UG base pairs. So they actually have a preference to, they like being aligned. So all the U's on one side and all the G's on one side. So this contrasting with the AU base pairing. So we can start making those base pairs. Okay. Okay, so this is again a more complicated structure, but here you see we have more base pair requirements this time. So keeping in mind that we have to have less than 15 CG base pairs, and I know that they're quite strong. I'm going to start by making as many CG base pairs as I can. Okay, and I think you can quickly appreciate that we don't have 15 base pairs, not enough to solve this puzzle. So we do have to use some AU base pairing, but I can finish by making more CG base pairs. Okay, and then for this part, we can start making AU base pairs. Okay, and you see we also have to have 6 or more UG base pairs. So we can make those, actually we'd have to change some of the CG base pairs. So go ahead. And you can see that sometimes when it doesn't satisfy the requirements, you can try and flip the orientation to see if that will work. It's a lot of trial and error with each puzzle. And so on. So are there any questions in the chat before I finish, or let's see? No, okay. So nobody had, well, I'll keep an eye on the chat, see if there are any other questions about RNA, and hopefully we can answer them before the end of the session. But now I can hand it over to Matt, who will talk about actually DNA.
All right, let me get the presentation going. I'll share my screen in a second. Okay. Share my screen too. Can everyone see what I'm showing here? Should see some nice colors, colorful diagram. Can you see me up? We're good? Okay. So hi everyone, I'm Matthew. I'm also from Mogen, and I'm going to be talking to you about another citizen science game. But first, we have to go over some quick background, and that is going to be about looking at the DNA of different species. So a really common thing that we do in this genetics field is actually look at the DNA from different species or individuals in a species and see how they compare to each other. Where are they similar? Where are they different? Tim already gave you a really good summary of DNA and the different bases. So there's 4 bases, and we usually give them each a distinct color. So you can see in this diagram here, each base is a different color here. And don't worry about the letter, just worry about the colors right now. So the reason we actually look at the DNA of other species and compare them is it helps us find genes, identify new diseases, or how those diseases that we do know about, how they might work, and how even related species might be. Well, how long ago did they evolve? Are they part of the same family? That kind of thing. And we actually do this through a technique called multiple sequence alignment. And basically, so what we have here is you take 2 or more DNA sequences and you try to first match up the areas that are similar, and that'll help you identify where they're different. So each row here, I'm going to show more, but one row of colors is one DNA string of letters from one organism. So we're going to compare a whole bunch, and you see where it goes down the same color all the way. That means that every organism has the same DNA there. So you see from this, though, we know what's the same. Where might there be a difference? And when you align them all like this and can know where they're the same, you can see, oh, these 3 right here, instead of a T, they have a C. So that means that here, these 3 organisms are different, their DNA. And this can tell us a lot about what's going on with the animals themselves. So the actual game we have here is called Philo. It's a game from McGill University. And Philo basically it can be very tricky. What it helps the scientists do is align the DNA. The DNA can be very tricky to figure out where to line it up to figure out where it's different, because it can be too different, which makes it very difficult where to find out where it's similar enough to match them up together. You have computer programs to help with that, but checking if the computer programs are accurate is difficult, and that's where you and Philo can come in. People are really good at seeing patterns, and that's why we can use citizen scientist games to help scientists make up or figure out how to align stuff. So with Philo, you're going to be given different DNA sequences that a computer already predicted are lined up, and you're going to confirm if it was right or not. And you'll be doing this by trying to match up the different bases. So each base is a different colored circle, and you're going to actually be able to click and drag these around to line them all up. And when they're aligned, you'll get points and your score will go up. And hopefully this data will help the scientists know if their computer programs are right or if they need to change them. So basically you'll have this and you'll be able to line them up, and they'll be different in some areas, but you're going to try to find areas that are similar. So I want to actually go through the game itself right now. And let me put the link in the chat. So I think I have access to the chat bar. There we go. So if you just go to this website, so philo.cs.mcgill.ca/play, you should be able to get to a screen like this. You're going to actually maybe be asked whether to make an account or whatnot. You can sign in as a guest. You don't have to make an account or anything like that. And by the way, if the music is whatever annoying, you can just right click and hit mute. So what I would suggest you do is you go into story mode first, okay? And with story mode, lets me go all the way back. I've been playing it to make sure we're going to go through the tutorial together, okay? It's pretty short, and then you're free to go and do what you like. So you don't have to click 1. It's like a little story. They'll give you a little bit of background. So you can skip that and start in at 3. So what you're going to have here is you're going to talk about what I just showed you in the PowerPoint is the different letters from the 2 different species of a rabbit and a human. So we're going to try to make them align. And as it's telling you, you can move them across. And when you move a circle, every circle beside that circle will move too. So you can't switch the order, but you can move them around. And when you match them up, that's great. You get points for that. And then you add another species, and you'll try to match them up to all of them together. So you want to match them up here. They can be different, but you want to make sure at least some areas of similarity are there. If they're not matching up, you will be penalized a point. If there's gaps, those are even worse. You do want to avoid having gaps. Those are more penalties. And then let's go through doing the first level together. So basically there's going to be a goal of points, and as soon as you get past that, you can move on to the next level. Okay, so here we go. I have these. We already have a few matched up, so I can click and drag, and you see how it actually moves them around. So you can move them around. Let's just drag them all. They're matched up, and great. So when this goes yellow and your score reaches the goal, then you're good to go. So you can click that, and then it'll add the next one, and we can try to match them up. And you can see it's a bit okay. Now we're good. We have a little bit of overlap. You can actually move them around, but we don't want that because we have gaps now. So it depends. Sometimes they're penalized more, sometimes less, but we have a bunch of matches. So either of these solutions work. And then we can keep going. So if you guys want to feel free and just play around with this too while I go through it, I'm going to continue playing. There will be a point if you go far enough, I forget which chapter it is, but they're going to actually ask you to, these will be grouped together, and you'll actually be prioritizing matching these species that are more closely related to each other over matching all of them. So they add all these other little things in the farther you go, but that's way deeper into the game that we wouldn't have time for today. But this data really is going to help scientists figure out, okay, are there computer programs doing the right thing when they're trying to match up the DNA? So feel free to keep playing here. And if you got, even so, you see I have a gap here, but that's okay because I have enough similar DNA. And we'll keep going. I'm going to match all those up. So the first few levels are pretty easy. They do get a bit trickier. And remember, you're not going to have perfect DNA sequences aligned. There's going to be gaps. There's going to be something that's different because species are different, so they don't fully match up. And then we can figure out where they will actually differ. And that kind of makes the species unique, where their DNA is different. And you can keep trying again and again with the harder ones, especially to get a better and better score. Whoops, that is chapter 6. I do not want to go all the way ahead that much. Let me go back to where I was. Let's see, this is complicated. It gets way more complicated the farther you go, but we're not going to get to that today. So let's continue on. I think it was 6 we were doing. So you'll actually also, if you need a hint, it'll actually help nudge you in the right direction. If you wait long enough, this little question mark will appear, and it will rearrange the base, a few bases at a time, into what might be some suggestions to what to look out for. So you can see right now, yes, we're minus 30. We want to get to 10 to move on. So let's just drag everything together. I kind of like dragging it all together and then messing around with it afterwards. And these two perfectly line up. Well, almost, except for that one spot right there. And we can keep going. We've reached our goal. Now this is actually going to have a little bit of difference here. We're past our goal anyways, but it will vary. Okay, we just made it. We could probably make this better. You can see our top score someone got is 44 by the end of this. We could actually look at moving them all the way here. Now that doesn't reach our goal yet. Well, barely, but what we're going to have is another species added. And it seems like it also has this very long stretch. So sometimes you'll get your goal, but if you feel you can do better, you can play around with it and get to where you're happy with your score. Believe me, when I was playing around with this for the first time, some of them are very difficult. So there we go. Awesome. Okay, it's bringing me over here. But so this one I didn't do too well on. Let's go back to this one. Maybe I can do better with everyone watching. And I'll actually tell you up in the top corner what kind of genes you're dealing with. So these ones, I might not know the letter, it's not showing you the letter, but what DNA you're working with is from the blood and the immune system of these organisms. And you can see down here we actually have our little diagrams of our organisms. We got here, looks like some humans, and that's a really small one. I can't tell what that one is. The other one is, but there we go, we're lined up. And yes, there will be sometimes gaps. Okay. So that's okay. Let's push this set. There we go. And it doesn't always fully align, but that's okay. And put those guys together. So those, nope, that's not good enough. I think putting it there, yes, we just made it. Now, sometimes you'll get a very little bit of DNA here, but that's okay. Just try to make it match where you can. So I got that. Got that. No, that's not really working. Okay. Doesn't like the gaps. See, I'm like, oh, what do I do? So If you give it a second, it'll tell you a hint eventually. So what are we going to do here? Well, maybe over here. So even if it doesn't fully run, you kind of want to try to have as many similar colors in one column as possible. So the reason that wasn't working initially was, yes, we might have matched the orange up, but you see there's 3 green here. There might not be a green here, but there's 3 green. So it likes when you try to match them up. So we can actually tell this organism is different here and here. All right, now we have more to do. All right, now we might be able to do a little better. And you can slide around previously matched stuff if you want. See what's going on there. Sorry, there's a loud noise of someone honking their horn outside of my building. So, actually, I can't recall what I'm supposed to do here. This one. Let's see, this one gave me a lot of trouble earlier. But does anyone have any questions about the game? I don't know if I can see the Q&A right now. I don't want to make sure I'm not missing anyone. So there's also, and I would recommend afterwards, if you want, you can play through specific challenges in the philo. And it gets very complicated because they'll introduce RNA, which will actually be a little bit similar to what Tim was talking about with its structures, because you'll have to actually take into account potential structure things, issues, as well as the sequences. So that's the RILO, I think they call it this. So you have plenty of different challenges as you go farther and farther. I got that aligned. I can't recall what they're asking for me now. I think it's the gap. Yes. See, they didn't like the gap that much. It's that penalizing to have a gap. And the bigger, usually it's better if you have to have a gap, by the way, have one big gap rather than lots of little ones. Lots of little ones is very much not what the game likes. Let's just play around with this. Okay, there we go. Didn't fully see, there's lots of differences here, but there's enough similar matches that's pretty consistent and we're good to go. So this one actually is even just too big. Sometimes, because you only have so much space to work with, you can actually push around everything afterwards. So let's go like that. Like that. Oops, too far. So we don't know, it's still not perfect, so we can still do better. And this is where sometimes you might need to have a few gaps come in. Potentially, probably not here right now. But that's essentially the gist of the game, is going through these. Now I want to show you actually with the different species. When they actually ask me to put together, I think it might be this chapter. So yeah, here. So there's a little tree, a little family tree that shows how related these ones are. So the closer together they are, the more related they are. So what they're going to want is the game will give you more points for making these two as match closely as possible. If this one doesn't fully match, you won't be penalized as badly as if these two weren't matching as close as you can. So let's get these all matching. So they're only different one spot. That's great. They're going to maybe ask you, sometimes they'll do one at a time, sometimes they'll do two at a time, and then they'll want to connect all of them. So let's match these two up. This is a very simple one. Perfect. And now we have to worry about matching all four. But you want to keep these two and these two as similar as possible while matching the families. So see, these might be quite matched, but these two are way too off. So we slide this though. Oops, a little too far. There we go. Now we get another two. And there we go. Now we got to match everyone together. So sometimes if you got, the way you placed it, you might have actually already gotten them all aligned, but it looks like we have some work to do still here. Start by dragging some around, seeing what we can get. So this is a little bit, oops. So we're almost there. We just need to match it a little bit better. Let's see if we can. Oops. There we go. So there's a big gap here. We have 4 of them actually, but that's okay because we had within each one, there's these 2 guys, we've kept them very similar. These 2 guys, we kept them very similar. These 2 ones we kept very similar, but also try to maximize across everyone. So this is where it really gets a bit difficult, and they're actually looking for some really good alignments. So usually there's one practice one, and now we'll go into an actual. Oops. Back at question chapter 6. It keeps bringing me back here. So I guess if you get stuck, like I said, there's the hints. Okay, so we have a lot of different ones. So again, blood and immune system, you get to try different ones. And when you get to the challenges, we can actually, I was about to say, the challenges, you can actually pick which part of the body you would want to work with: immune system, digestive system, heart, stuff like that. Yeah, so let's keep going. We've matched these guys up. Now let's match these 4 up together. Okay. There we go. We don't have these fully lined up. And this is where, you're looking for patterns. Okay, no. Maybe this one. Okay, we're on to something. You can do a little bit at a time to see. There you go. All right, now we'll read this 3rd one. Let's see if we can line this one up. Maybe I think this one will give you a hint. If you wait long enough, okay. Maybe you're a bit stuck, like that, and it'll nudge you, like I said, in the right direction. So if you may think about putting a bunch of ones apart, it's not great to have gaps. It really isn't. You can play around with that. Okay, so this one I'm actually not too sure of right now. Let's see a hint. So you can push it all the way across. So I just want to make sure you guys realize also like how you can't rearrange it or anything. Just make sure you're pushing it across. Okay, so we only have a little bit here. So let's try our best to align this. No, doesn't go there. So if you guys look for pattern, these 4 right here. Perfect. Now the big one is to align everything, which is always a blast. So usually you'll end up, because you see farther and farther more distant related from each other, you'll probably have more differences between this group of 4 and this group of 4. So let's see what we can do. I think I see some stuff that lines up over here. Okay. No, it's not yet, but I see purple and green. I was looking here, purple and green are lining up. There we go. So now let's try these ones. So we're only doing these 2 now, so we're not yet aligning everything. And I'm going to stop in a second once I finish this. But actually, before we continue, nothing in the Q&A right now, but if you guys have any questions about anything we've talked about today, we're all happy to answer them, or if you have any trouble with a specific game. And you're more than free. You can play this on your own time. You're actually, that's the whole point of these games, is try to get really kind of everyone, as many people trying different things out and seeing what works. And that's how these citizen scientist games work really well. The one that Kiki shared reached tens of thousands, hundreds of thousands of people and participants, and millions of experiments done, which really helped the scientists actually put together a good program and tool to help them in their specific fields. And it's through this effort, it makes, a small group of scientists can only do so much in so much time. Sharing it with everyone else and getting everyone else to put their input in really speeds up the discovery process. You can really contribute. So I'll just hang out here. I think we'll be done in a couple minutes. We'll keep going. So what will we do here? Maybe get some stuff aligned there. And if you're not sure, they're going to have this guy looks very different than the others. And you're trying to get these guys aligned. Let's see if we go all the way to the end. There we go. See, we got the orange all the way through. Those are kind of the best ones when you have a base, a DNA and color that every organism shares. Yeah, this lets you appreciate how different they might be. And we could do better. Someone was able to get 281. So there is a better way to put these together. We just hit the minimum. Awesome. So level complete. So yeah, that's the game. And if you want to try other ones, so this is more complicated. And this is one that you get near the end of the game, which will be arranging RNA. I won't go over that today because it's a bit too complex right now. But here, Philo, you were to click, okay, you want to do the digestive systems. This is something to do separately and not part of the story mode. From infectious diseases, something with your metabolism, cancer, the heart. So we could do one from the heart, for example. We're working with DNA from the heart. I think this might have been one we'd already done, but that's okay. So not fully matching up. And I think, considering our time, we're almost done. I will stop sharing, but I hope you guys learned a lot from this. And I guess I'll give Kiki hand it over to Kiki for the outro. But thank you so much, everyone, for listening. And hopefully you had some fun trying this out.
Yep, if anyone still has any last minute questions, this would be a good time to ask. All right, guess it's a no. So we're gonna just pass it off then to our next Mojang booths. I think the Science Rendezvous hosts will be giving an introduction, so I'll just stop here, I guess. Thanks.
Yeah, thank you guys so much. That's super cool getting to see the whole citizen scientist sort of getting an introduction to that. I know even that term is not something that I knew until maybe like last year. So I think getting everybody to kind of get involved in sciences is definitely super cool. And getting to have all these games that are designed to really help educate and let people try out how genetics is kind of working on the most molecular level, definitely super cool. So thank you all for that. We will be moving on to our next session for molecular genetics, which is going to be the DNA extraction challenge. Yeah, I'm excited to see what this will be. And whenever you guys from molecular genetics are ready, the floor is all yours. And for all of those participants, once again, please, whenever you have a question, add that into the Q&A section. And when we have time, we'll be able to take those on. All right.
Hi, I'm supposed to be sharing slides next, but I don't think I have the ability yet.
All right, we will definitely fix that. Let's see.
So I think I just got it.
Wonderful.
Get you guys all set up. Make sure you can see all my slides properly.
Yes, that looks good on my end.
Fantastic. Okay. So today I want to welcome everyone to the DNA Extraction Challenge. I'm going to give you a short introduction about DNA, and then we can walk through a really fun experiment together that you can also try at home. So we're made up of trillions of cells, and each cell has its own job, kind of like humans do. Some cells help us detect light and see, others help us touch and feel things. They also make up our hair, our skin, and our muscles. So there's around 210 types of cells in our body, each performing a different job. But how do these cells kind of know what they're supposed to do? So they're actually told what to do by a special molecule called DNA, which is actually short for deoxyribonucleic acid. And each person's DNA is unique. We share a lot of our DNA with our biological families, and it's actually passed down from parents to children. So your DNA is a combination of the DNA from each of your parents. And a fun fact is if you took the DNA from every cell in our body, which we have about 37 trillion cells, and we stretched it out, lined them up end to end, you would be able to circle the Earth nearly 3 million times. So we have a lot of DNA in our body. And what does it actually look like? So here I've shown you a strand of DNA, but it's a very blown up version. You can't see this with the naked eye because DNA is actually very, very tiny. It's only about 2 nanometers wide. To give a little bit of context, a human hair is 100,000 nanometers wide. So scientists actually use very, very powerful microscopes to be able to see DNA. And under the microscope, it's actually just a really long kind of string, but through various methods, they figured out that DNA actually has a very distinct shape, which we call a double helix. So this helix is made up of a backbone, which is shown here in purple, that gives DNA its structure. And then the letters of the DNA alphabet, of which there are only 4, A, T, G, and C, they're also called nucleotides, are used to write the instructions carried by the DNA. So only certain sets of nucleotides can fit together, so kind of like puzzle pieces. A only associates with T, and G only fits together with C. All the instructions that are needed to tell the cells what to do are written just by organizing these 4 nucleotides into unique combinations, kind of like words. And then these words are put together in unique ways to create unique sentences. And these are the instructions that are given to the cell. So if the host can actually bring up poll number 1, we want to kind of ask everybody or quiz everybody what has DNA. So the options we have, here are all living things have DNA. Do we think rubber ducks have DNA? Do non-living things have DNA? Or am I all these options wrong and none of the above have DNA? So I can give you guys a second to answer. I think we have about half of our participants have answered so far. And then maybe I can give about a 30 more seconds to kind of give everyone the chance to participate. We're at about 2/3 of our participants. And that was about a minute. So I think I'm gonna close it down now. I'm gonna give 10 seconds just for anyone who wants to throw in their answer at the last minute. So 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. We're gonna end the poll. I'm not sure if you can see the screen poll results or if they'll come up, but it seems like everyone has chosen all living things, and I'm glad We're kind of all on the same page here. You guys have a really basic knowledge of DNA already. So that should make today really fun. Let me close this. Yep. So how do we test if something has DNA? So if we wanted to figure out if your dog had DNA, a strawberry, a chocolate bar that I've shown here, or maybe a cotton ball, how do we do that? Do we just mash it up and look at it really closely with a magnifying glass? So today we can actually use common household items found in our kitchen to extract the DNA. So today we're going to perform a DNA extraction together on a strawberry as well as a piece of chocolate. So Ryan and Yi-Ching are joining us from the lab and will walk us through all of the different steps. So if you can actually find the materials, you can follow along kind of right now with us at home, but we'll also be posting a link to instructions. I think it's a PDF file that you can try this on your own after the event today. So with that, I think I'll pass it over to our friends in the lab, Ryan and Yi-Ching.
Hi, everyone. Testing.
Can everybody hear us?
Yep, you're all good.
Okay. Just want to see if we can share.
Did you guys? Okay. Yeah, you're good.
I can see it.
See it. How do we make it?
Are you guys having issues with slides? I can keep on sharing the rest of the slides as well.
No, we don't really have a slides. We just want to make our screen as big as possible.
Oh, you're sharing your screen.
Stop sharing your screen.
So,
okay. I got you.
All right, there.
Yeah.
Everybody can see us, and you can hear us. Okay.
Cool.
Excellent.
So welcome everyone. So joining us is from Department of Genetics at University of Toronto, and we have Ryan Marks from Sick Kids Hospital, and my name is Yi-Ching, and I'm from Mount Sinai Hospital. So we're here joining from Mount Sinai and in downtown Toronto. Hello everyone. So what we're going to do today is to extract DNAs from 3 different sources, and hopefully we can answer the question that just now Emily has posed. So our first candidate is strawberry, which we can see is large and sweet. The second candidate is a piece of chocolate, okay, which in this case we code this possibly with question asked if we can ever get something out of this DNAs. The third is actually ourselves. We're going to actually do our salivas, and we try to actually regurgitate our mouse and see that we can actually get some cells out of our DNAs out of ourselves. This I hope will be also a good Mother's Day present for your parents as well. So let's begin that. So we'll start with
So
Ryan.
if you want to follow along with your PDF protocols here, the first step that we're going to do is to take the fruit or whatever it is that we want to look for DNA, and we want to smash it up into really small pieces so we can get that DNA, if there is any DNA, out of the cells. So the first thing you're going to do is put your fruit or your sample into your Ziploc baggie. And what I'd like people to do is close it up and just kind of smash it around and make it really kind of mushed up.
So in parallel, I'm also going to take a piece of chocolate, and I will say take maybe a 1/2 centimeter times a 1/2 centimeter big, and do the same thing as what Ryan is doing, and that's what I'm going to do. And I'm going to put in a Ziploc bag as well. So we're just crashing it right now. Ryan is easier, for me it's slightly difficult. I got the chunky piece to be broken up.
So when your fruit is all mashed up, it should kind of look a little bit like it went through a blender. It should be nice and liquidy. If there's chunks, they should be a little bit like really small pieces.
And I'm just about the same, like this.
All right, so the next thing we need to do is make a solution that we call a buffer. And what these solutions do is a few things, but one of the main things it's going to do is take the cells and and basically break them open so that they can release the DNA that's inside of them. So there's instructions on how to make that buffer, and we're going to make it here along with you so you can see. So actually, if you could pass me the water there. Perfect. So the first thing we need to do is to mix
tap water. We got tap waters here, or anything from a drink water bottle, doesn't matter. We just want to prepare the solution.
Perfect, thank you. So we want to add a half a cup of water, which is about 125 milliliters. So this 50 milliliters. We'll do another 50 milliliters. And we'll add another 25 here.
So the amount is approximate. You can just roughly measure by a half a cup.
Yeah. Perfect. The next thing you'll see on your protocol list is we need to add some dish soap. And the dish soap is basically going to help, soap is like a fat, and it's going to help dissolve the fat membrane around the cells. So this is what's basically going to dissolve the cell and release the DNA into the buffer that we're using. So what you're going to do is take any dish soap is fine. You're going to take 2 teaspoons of dish soap and add it to your water. So there's 1 teaspoon. And here's a 2nd teaspoon.
Okay, so while Ryan's preparing the solutions, we propose our first challenging questions, and as to that, why do we actually need to use the soapy solutions in here? And and if anyone can think about an analogy as to why we are doing it, and by thinking about where is our DNA and in the first place, and how do we actually get the DNAs out of the materials, which is the strawberries in this case, from. Okay, so we are now having our Ryan's buffer made, which is pretty much the soapy waters that are being made. Okay, so the next step is that we're going to add salt to the solutions as well. And what we're going to do is Ryan is just now scooping 1 teaspoon of salt, and we're going to add this to the same solutions just now we have prepared. So right now the solution now has half a cup of water and also 2 teaspoons of soap and 1 teaspoon of the salt in the solutions. All we want to do is just swirl it a little bit to try to mix them well. If there is bubbles, that's okay, but let's try to minimize the bubbles that are being made. While you're thinking about why the soapy waters is going to be useful for this purpose, think about why we actually are thinking, why we are actually seeing the bubbles showing up. Okay, so I think now the solutions are prepared. We're going to add the solutions to the materials that we have just prepared already to smash out. This step, we are actually going to get the DNAs out of the material that we have just prepared. Basically, get the DNAs out of the strawberries. And if I ever have any DNAs in the chocolate, we're going to get this out of my chocolate as well. Okay.
So in the protocol, it says to add 2 teaspoons, of this buffer solution, but we used kind of a bigger strawberry. So we're just going to do a rough estimate here and add some of the solution directly to the bag. This will help with the next step as well. Okay, perfect.
So if you ever wonder, we can get a small cup like this, dollar store cup, and we can get like 1 cup or half a cup of the solution, pour them into here. Since my chocolate is way smaller than actually about half the size of Ryan's strawberry, I'm gonna get half the solutions as what he has done. So I'm gonna get a full cup of the soapy solutions he just prepared.
All right. So now what we're going to do is just help lice up all those cells with our buffer solution. So we're just going to mix that soapy solution all around with our samples here. First there. Yeah. Okay. So the next thing we need to do is we have this mixture of our buffer, all the lyse cells that we just broke open. And if there's DNA, we should have some DNA in the solution. So now what we need to do is filter out all of the, the leaves from the strawberry and any of the fiber from the strawberry that hasn't dissolved into the solution. So what we're going to do in step 4 is just use any household coffee filter that you might have for your coffee machines. And we can put these inside. We're going to use these special little tubes that we use in the lab all the time, but you could put this just into any cup or any coffee cup maybe that you have at home, and it should be fine. So we'll take the filter. And we're going to put it into the rack or into the tube here. What we'll do first is we'll just pour the solution right into the coffee filter. And you can see how all this soluble parts will just drip through into the tube. You can see now that the chocolate sample that we have is dripping through. I'll give that to you. And you can use a little bit of force and you can squeeze some extra fluid through the coffee filter as well.
We'll do the same thing also for the strawberry as well.
Can you have another tube there?
Oh,
So we're just setting up that filter again here. And if you don't mind holding that for me. So what we'll do is we'll pour all of the strawberry solution into the filter, and we'll filter out all of the material that is not soluble. We'll squeeze a little bit of it through. Excellent. We'll do a little bit more.
Just slide it.
Yeah. All right. And you can see we didn't pour all of it in, but this should be more than enough to get some DNA out of the strawberry if there's any in there.
Awesome. Great. So now we got the chocolate, we got the strawberries in it, and we are now going to get a clean mouthwash cup, and we're going to pour out 1/3 a cup of what we have just obtained from there. Okay, so Ryan is going to get the strawberries. I'm going to go with chocolate. So he's going to pour out 1/3 a cup of the solution. Same thing. Okay, so if there's ever any DNAs existing, and it's going to be inside of those solutions. So the next thing is that we are going to actually try to visualize the DNAs and try to get the DNAs out of the solutions. This, we are going to use a magic solution called rubbing alcohol. Okay, so right now the solution, the DNAs is actually dissolved amongst any other things in the solutions we have just prepared. By adding alcohols, we are going to create an interface, whereas the alcohols will stay on top and the chocolate or strawberry solutions, water solutions will stay in the bottom. Right in the middle, that will be where we're going to see the DNAs. So since we have 1/3 a cup of the strawberry or chocolate solutions, we're going to pour about 1/2 a cup of the rubbing alcohols into it. If you don't have a rubbing alcohols, try to find your cabinet. If there's any vodkas or high content alcohols, do not use beer, by the way. We try to use alcohols that are really of high percentage of alcohols in it. So rubbing alcohols will be your first choice. If not, vodka will be doing the trick as well. All right, so now Ryan's gonna pour it into the strawberry part. Okay, so if you you could see now we have the solutions like this, and we can hold it like that. So we can have two separate layers, okay? And the top layer, the either transparent layers will be where our alcohol is, and the bottom layer, which is our strawberries, and this is the pinkish layer. So what we want to show you is at interface, you can see there is all this bubbly and sticky stuff. This is exactly where your DNA is. If you use a toothpick to come in, okay, and you start picking it, this will be where your DNA is. Can everyone see where our DNA is? This little chunky stuff. Maybe perhaps give me a clapping hands if you can actually see the DNAs. Okay.
I'll put it inside the profile.
Yeah, I'm going to. Yeah. So it might be a little bit difficult to actually see it and preserve it. If you still have a little bit left over rubbing alcohols, we're going to pour more rubbing alcohols to another cup, and we're going to permanently actually preserve this DNA. So what we're going to do is to use a toothpick again, okay, and take the DNAs into the rubbing alcohol, okay, and this might make it slightly more visual, okay, if everyone can actually see where my toothpick is, this whole chunky stuff, that's where your DNA is. Okay, I'm going to do the exact same thing for my chocolate solutions. And let's see if magic also happens like that. So Ryan is gonna pass me the rubbing alcohol. We're pouring again into my chocolate solutions. And we're going to do the same thing and try to see if a toothpick can actually get anything out of it.
So before we take the toothpick out, we're going to ask everybody to try to make a prediction for yourself if you think that we're going to be able to find any DNA in the chocolate.
Yeah, it's actually hiding where the beaker, where my cap is, and let's see. And 3, 2, 1. Let's see if there's anything actually coming out. Okay, I try to use my toothpick to do it. At least there isn't really things so far. Can anyone actually let's do a poll and see that we're expecting to see anything. Okay, so right now we got about 50-50 split. Cool. We have 5 seconds, so this gives me actually more time to to see if there's more DNAs. Okay, and it does look like right now we have a perfect 50-50 split right now. And so from our end, though, we actually do not see at least as much DNA coming out of the chocolate solution. And in fact, our prediction at this point is that there is no DNAs coming out. So just to actually try to see if I can actually convince you, I will do the exact same thing, following exactly what Ryan has done just now. I try to squid something out of it, let's see, and then dip them into the rubbing alcohol solutions. Let's see the comparison. It's clear. So from my side, I really don't have, actually, I don't have anything on this. And Ryan has a big chunky piece of DNAs in it. Okay, so the answer from our side, the response from our side for now is that the answer is no. So there is no DNAs from the chocolate. Okay, and so for those of you who actually chose yes, we will talk about this after in the discussion sessions. We'll talk in more details as to how this experiment was performed and whether why there is no, we think there is no DNA in the chocolates we have gotten.
Do you want to do the cheek samples?
Yeah. Okay, so we want to know if there's, first of all, any questions till now. And if not, we're going to perform one more experiment, which is to actually get DNAs out of our cells. And I guess just now we answered the question as to by Emily, like, so where can we actually have the DNAs? All living things has DNAs just now. And so strawberry is a plant. So it has cells and it has it's a living things. And viruses, even if it's not a living thing, it still could have nucleic acid, either DNA or RNA in it. We can also extract something out of them as well. Us, human or mammals, or animals in general, we are also living things. We're also made up of cells. And as such, because we have cells, and there is a big chance that we are going to get DNA out of us. And this is what defines us who we are. And this DNAs will shape the future of us since we were born. So what we're going to do is to actually get the DNA out of ourselves. In fact, the DNAs inside our body are almost exactly the same across the entire body in ourselves. So because of that, we're going to take the part that is the easiest to obtain the cells, which is through our mouth. Lowering my mask, we're going to actually get the same cup. Okay, we're going to get some salty waters. So in this case, we'll try some either Gatorade or put 1 tablespoons of your salt again, and dissolve it in about half a cup of solutions of waters like what we just did. And so we're going to do that right now. And some water. So I'm going to scale down the amount just as if like I was to do in this 100 mils. I'm going to scale down a little bit and add a slightly smaller amount of salt. Thank you. The next thing we want to do is to actually don't drink it, just swallow it, just put it in your mouth and start
swishing.
And I'm not going to be able to talk after that. So we're going to do 2 minutes. Let's do a very good sorrow squishing. And where possible, chick is where our cells are mostly coming from the mouse. And there's also a bunch of bacteria inside of mouse. I wish my mouse is as clean as possible. And so where possible, you can also use your tongues to try to rub against your chicks. So try to get as much cells as possible. Let's do this together for 2 minutes. 1, 2, 3.
Perfect. So while my partner's swishing, I'll just kind of reiterate where you're getting the cells from here. So in your mouth, when we're swishing with this saltwater solution, we're collecting some of the dead cells on the inside of the cheeks, the tongue, and as well, there are a lot of normal, healthy bacteria that can be in the mouth as well. And we're collecting all of these sources of living tissue or living cells. So the idea now is that unlike the strawberry, where we're going to smash it up and break the cells apart, we can take that same idea and use it to extract DNA from our own cells. Perfect. So after you've swished and you've, gotten lots of material from your mouth here, you're going to basically collect that sample in a little tube. Excellent.
So there we go.
Perfect.
Finally, I can talk.
Yeah, so in this solution here, we should have a bunch of, you're going to want to make sure your mouth is clean, no food or things like that before you try to do this. But we're going to have basically a bunch of different types of cells from the tissues in our mouth and bacteria and things like that. So the slight variation is because there's obviously a lot less material than when we were working with the strawberry, you're going to just adapt the protocol a little bit here. So we're going to add just a few drops of soap directly to the solution that contains the cells from our mouth. Doesn't have to be exact.
So 1 teaspoon is more than enough.
Perfect.
So yeah, if you have the toothpick, you can feel free to swirl it as well. just consider that we're doing all those experiments in parallel, like what I did just now with Ryan, and very similarly, we have the solutions has the exact same component, which is the salt, which we just add. And also we have the material, starting material, which is my cells and salivas. But more importantly, we also have the soap altogether. So all the three things are there. The next thing is we are going to add rubbing alcohols to it. So we already have the rubbing alcohols here. So we're going to start pouring them into the solutions. And it works the best if the alcohol is actually chilled. If you can put the alcohols in the freezer or in your fridge, and that will actually help even better. So we're going to start adding it. The amount is about 1 to 3. Okay, so we need 3 portions of your alcohols and 1 portion of what you have just poured in. So if we could see at this point,
it might be difficult to see on the screen, but I'll just kind of describe what my partner's doing here. So just like you'll see those 2 phases between the alcohol layer and the layer of salty water underneath. If you kind of pick the toothpick around where those 2 phases meet, you'll start to see these little stringy pieces kind of being pulled through the solution. Now, like I mentioned earlier, the strawberry, there's a lot more material in here to collect DNA from. So you can see that that directly correlates with this experiment we're doing. You can see just how much DNA, all this little dangly bit on the end of the toothpick that we collected from one giant strawberry compared to the very few cells relatively that we collected from my partner's mouth and whatnot. And now if we take the sample and we just store it in a little bit of pure rubbing alcohol, might be able to kind of compare how much we got from our cheeks versus the strawberries.
Just show them. This is where we have what we have. You can see the string, the bubbly thing, that will be your DNAs. Okay, then I'm going to transfer the thing to the rubbing alcohols. This will be our final product, which is your DNAs from your mouse cells, your chick cells. Cool. I think this is it. That's our extractions from materials, different materials. And at least we know that from our mouse cells and also from strawberries, we can get DNAs out of it. And as for ourselves, and I think that will be a good present for your parents as the Mom's Day, Mother's Day, and Father's Day that's going to come in a week and in a month.
We've actually got a Q&A if you want to answer it live. Sarah asked, is this how they take the PCR samples for COVID testing?
That's an excellent question. So the PCR, it's exact same principles, except for that we are doing an automated process. And just it's a little advertisement. This department and also Mount Sinai Hospital take care of more than 50% of the COVID testings in this province. And a big shout out to our colleagues at Mount Sinai for and all the laboratory medical technologists for keeping us safe and by performing this hard work. To answer the question, is this how we actually are doing this? Extracting nucleic acid is a very similar principle in this case, and except for that we're not actually taking the chick cells in this case, whereas mostly we're taking the cells from our noses. And viruses, however, when they are either inside of our cells or actually being secreted into the airways, we're trying to get our samples from there. And very similarly, we're trying to use solutions that actually not contain soap, but contain a very similar material that will actually do the same trick. They will try to lyse the cells or lyse the material that are actually capturing where the nucleic acid. In COVID's case, it's not DNA, it's actually RNA, but they are of a very, in terms of our purposes, they're of very similar properties. We can extract them out. And I guess the big question to follow up is that what can we actually do after we get all these DNAs? So the DNAs we got is something called a genomic DNA. We got the really crude, low quality samples, DNAs out of the samples. So the question is, what can we actually do in the next step? And the application, it's a ton. So we can actually perform forensic analysis. The materials we have is very tiny. The first thing probably we want to do is to amplify this material, so then we can actually perform adequate analysis. This is exactly what the COVID testings PCR is actually doing. So the virus's material is in very tiny amount. We can then actually target it to amplify that material and see that, and then get a conclusion if there is actually detectable material in or not.
Yeah.
So.
And yeah, I guess just to reiterate that, the biggest difference between what we do in the lab and what you can do at home is it's just a higher quality reagents, more pure, and the product that we're extracting here is just very crude. So there are some additional proteins and other molecules that bind with DNA that are still likely bound in this case. So you can clean this up and make it more pure. But yeah, for all intents and purposes, it's a very similar procedure. Not sure for the host if there's another question in the Q&A there.
Nope, you're all good. You've answered all of them.
Excellent.
Okay. I guess the next point we want to actually raise is that what Ryan and I did side by side just now is it's a science principle. It's called a well-controlled experiment. I can't say it's a really well, fantastically controlled, but we try to model this principle at least. Whereas if we want to actually conclude if there's anything differences between the two, we have to do exactly the same thing, use the exact same solutions, follow the exact same process, and make sure there should there be any differences, then we can at least propose the differences between what we actually see is due to the only thing that is different between the two. So we hope this also is a kind of a scientific thinking that you can actually thought about when you are actually in the future to perform your own experiments.
So we actually have, sorry, we just have a quick question in the Q&A that maybe you guys can address. Someone is actually asking if we could show these samples under the microscope. I guess for our purposes today, we can't, but maybe you could describe what that might look like.
Yeah. So I think in this case, so one thing that we do a lot in the lab is we want to visualize what kind of DNA that we have, or for example, like Carter was mentioning, if we've amplified a piece of DNA, we might want to see how big it is, for example. And some things we can do is we can run it through these special, they're basically bits of gel, and we can run it and we can stain the DNA. And we use different compounds to do that. Some of them, one of the most famous ones is called ethidium bromide. And these are fluorescent molecules that will basically, you can imagine they bind to DNA really well. And when you shine ultraviolet light on them, it makes the DNA glow. So we can see this DNA here because it's all clumped together. Remember earlier in the presentation, we talked about how DNA is like 100,000 times thinner than a human hair. So you can't really visualize it that well, but we can very easily visualize it when it's all clumped together. Remember we said we had, you know, a massive amount of DNA, and it's really quite impressive what the cell does and how it packs meters of DNA that's very, very thin all into this tiny little part of the cell called the nucleus. So it, you know, if you clump it all together, you can see it with the naked eye, but if we were to stretch this end to end, it would be something that we can't see. But to answer your question, there are some special stains we can use, but unfortunately with this kind of format, it would be hard to show you that.
I guess we would also add to this is like kind of generalize your question is how do we actually visualize the DNAs we are actually getting? So what Ryan mentioned just now is to use a special, some properties so that we can indirectly visualize where the DNA is by actually adding another thing that will bind to the DNA so that we can, by detecting the things that we add to bind DNA, we know then there is my DNA. And in terms of whether we can see it under the microscope, unfortunately, the light microscope we're using in the laboratories is unfortunately very, it is beyond the limit, the size limit that we can actually see it. And I guess at a later time, we probably will hear about like why this is impossible to actually use a light microscope to see small things like that, because there is special properties of the light, and you're going to actually causes refractions and diffractions. And so to get around with that, there are many actually different properties of microscope, and which do not actually rely on a light as the source. We can actually use electron microscope for examples to do things like this, and also X-ray crystallography to actually do these things. This is exactly what people have been doing in the 1950s when they first actually discovered the DNAs. So that's, hope that I answered the question. And I think if there's no questions, we just gonna wrap up what exactly we have been doing. And just to start off, and we have 3 materials, and this is very easily reproducible at home. And we have strawberries, that's one. And we have starting material, which is chocolate, and it was a chocolate, and it was chocolate. And we also have human sources as well. And where is the DNA residing in? That will be found in mostly in the nucleus. And although there are also some DNAs found somewhere else inside of the cells as well. But to answer the question, the most of this DNA is found inside of the cells, and primarily in the nucleus. And that's where we should exactly focus on to get our materials from. And that brings up to the magic solution Ryan has prepared. And the reason we add salt and also the soap is to try to actually break the membranes or the circles that are actually confining my DNAs, and in such a case, and we can then help to actually bring out the DNAs into the solutions, which we can then carry out the subsequent steps. And lastly, we add, because when the DNA is escaped or obtained inside of the solutions, we can finally then add alcohols. In that case, we can then use the alcohols to pull out the DNAs. Some questions from the Q&A will actually help us to extend the discussion on what to do with the DNA and how to visualize the DNAs. For your practice, and tomorrow morning, for example, when your mom or dad start preparing smoothies, and this will also be a really good starting materials for you to do that. If you actually get the smoothies, chunk of the smoothie, the clear part of the smoothies, and you add some soapy water into it, and also add rubbing alcohols to it, you most likely will see exactly what we have seen today as well. So I think that's concluded our part of the bench work part. And I think the very last point I want to make is that science is great, and molecular biology is the best. And we hope you consider science and biology in your university studies in the future. And signing off from Ryan and Yiqing from Mount Sinai Hospital in downtown Toronto, Molecular Genetics Department of University of Toronto. Thank you.
Thanks, everyone.
Wow, that was so cool.
The floor is still open. We can.
Yeah, if anybody has any
questions,
remaining questions,
we're still here.
Yeah.
Until the next session is here.
Yeah. Well, Ryan and Yiqing, do you guys want to speak a little bit about your research? Oh, I know we have an attendee who's raised their hand. So if you raised your hand, if you want to please put your question into the Q&A. Unfortunately, we won't be able to give you mic access for privacy reasons, but yeah. So everyone is asking, why did we not find DNA in chocolate? I think Emily and maybe somebody else also answered a few of these in the Q&A, but if Yiqing and Ryan, you guys want to give just a quick reason as to why?
Yeah, so this is another science attitude we want to have, is that you could prove there is. It's really hard to actually prove there isn't to begin with. But this is to be scientifically accurate. But to address this question, I want to say that I can't say there isn't any DNA, because chocolate is actually made from cocoa beads. And this was at one point a living thing as well. They're made up of cells. The question, however, I want people to think about is that the quantity and the process that we have been doing it. So yes, we have been crushing the cocoa beads into pieces, just like what Ryan has started with the strawberry. And after that, we started adding different materials, like try to mix them throughout this entire processes. And the amount of the cocoa beads remainings has been really little. And in fact, inside of your chocolate, those are that being sold in the supermarket, the amount of the cocoa inside of it is very tiny. And so in that case, cocoa beads itself has the DNA, but the final product, the chocolate itself, because it's no longer living things, and it does not have a DNAs into it. And that's why just now I was hesitating to say there is absolutely nothing, but rather there is, at least quantity-wise, is very small compared to the strawberries. And I guess that brings up another question, is why strawberry is so awesome for doing it. And in that case, Ryan and I just now were joking about the analogy of, if we want to do a survey as to like if school-aged teenagers actually like to do science, we have to find where to find a large population of our children, teenagers. The best place will be go to the school where there is a lot of students in there. You get a better result compared to if you go to a smaller school when there's only a few students. And the reason we use strawberry is because it has a lot of DNA into it. And in this case, we probably heard about this also in school, is that it has so many copies of the DNAs. In fact, it has 8 copies of the genetic material, whereas we only have 2 copies of our genetic material, and strawberry has 8. So if in fact, actually elephant has and other animals, you can actually check yourself and see how many copies of them you have. And this is interesting in the sense that the more the copies of the DNAs you have, the better quantities you will eventually get out of it. So that's the reason we actually chose strawberries. Any kind of berries in this case, I think will do the trick, and also bananas will also be a good candidate for that. I hope that answered the question.
Perfect.
Yeah, I think we have maybe just one more. Yes, just one more question.
I see that. So why would DNA be different in an organic versus conventional strawberry? So I think what you are referring to by organic is maybe like at the supermarket or grocery store, you see things labeled organic versus just kind of the general strawberries. So would the DNA be different? Well, the DNA, like we said kind of in our slideshow, DNA is the instruction manual that tells the cell what to do. And if it's an organism like a plant or an animal that has many cells, how those cells can all build up and create, you and I, or the strawberry plant itself. So if these strawberries, if they're, for example, different strawberries in different parts of the world, and they're different species of strawberry perhaps, or different species of plant, for example, those will have different DNA. As far as the supermarket, usually when they're saying something is organic, it's referring to maybe that it was grown perhaps like without pesticides or anything else. But if it's the same species of strawberry, it should have the same DNA, more or less.
And just to add to Ryan's comments, I fully agree with Ryan. And also, like, just a challenge for you to think about, how do you figure out if the DNA is the same or not the same? What kind of technique you could do afterwards? that brings back to our question just now, what can you do with the DNAs? So, and in fact, if you have 2 strawberries, I don't know, from somewhere like you take from China, and another strawberry you take from Alberta, and then you from a greenhouse from Alberta, and you bring the 2 together, and can you see they have the DNA, like what we did is exactly the same, but can you actually trace, are they coming from the same place, or they're the same species or not? So that is another way of answering this, to think about how do we actually do this.
We can use different, you might hear about sequencing technology. So we can use special techniques and methods to actually read the code of the DNA. So you might have heard that there's 4 different letters that we use to symbolize DNA. There's A, G, C, and T. And we can use special technology to actually take these strands of DNA, process them through a machine that can help us to determine what is the order of these A's, G's, C's, and T's. And that specific order is the instruction manual, you can imagine, that is telling the cell what to do. So if we wanted to say if 2 species were identical, we could sequence both of their genomes, and we could look and see what base pairs are different between, 2 different maybe species of plants, or, things like that. And that's actually the principle used behind some of these tests that you might see, like 23andMe or these ancestries, where they're looking at different base pairs between people from different geographical regions, and using that to say, where is the ancestry of your DNA coming from? So that's a good application to some popular products right now that are being kind of sold commercially.
That's why you're also 23andMe, you're also sorting.
And they also, yeah, you're going to collect the same cells from your cheek as well. Very similar process.
Okay, this one, yes. Great.
All right, perfect. Thank you guys so much for taking the time to answer all of our questions, and getting to see you guys do one of these sort of experiments that you can do with household products. Always super cool. Yeah, so we will be taking a quick intermission break just now. I know we have one more presentation from the molecular genetics group as well, but if everybody wants to stay on and ask a little more questions, definitely we can do that. Or perhaps at the end, if you have any additional questions, we can reach out to our presenters and see how we can get more answers for them. But yeah, so we'll be back here at 2:10, and in the meantime, if you guys do want, I will be putting a link to our Sci-Art gallery. So this is just a collection of the integration of science and art, and we've done it in a virtual sort of galleries type of view, and it's super cool. So if you have the chance, definitely go check it out. And yeah, okay, so we will see you guys at 2:10, and thank you everyone again. All right.
Thanks for having us.
We're really excited to show you some glow-in-the-dark model organisms today. Before we get started, we would really like to know if anybody has an idea or a guess at what a model organism is. So if you have a guess, or please write in the chat and I will read out your answers. So we're going to be showing you something that is glow in the dark, but what exactly is it? I will give a few moments for answers, and then I will continue with the presentation. Okay, can we go for the next slide, please? Thank you. So when we're talking about a model organism, we're talking about a living creature. That's what's meant by organism. So we have some examples up here on the slide, and a lot of these might be ones that you recognize from your everyday life. For example, you might see some fruit flies flying around your kitchen, or when you're digging in the garden, you might find some worms. A lot of the time, scientists also use mice and fish. And we use these living creatures, these organisms, as models. So what does it mean when we say it's a model? You might be thinking about a fashion model, or maybe you've heard somebody say that you need to model good behavior. And in that example, it's saying that you're going to be an example of something. And that's what we mean by a model organism. These are showing us an example of something that we as scientists want to study. So model organisms are living creatures that help scientists answer questions, and we can use them to try to discover how living things work. They also help us to understand lots of diseases, and hopefully we can use model organisms to learn how to treat and even cure diseases that humans have. Can we go to the next slide? So here's an example of a glow-in-the-dark worm. And this is a worm, like I said, that you could find in your garden, although it is very small, so you probably can't see it with the naked eye. And also, if you find it in your garden, it's probably not going to be glow in the dark. But this worm, which is called a C. elegans, it's very useful. It's a useful example because it progresses from an egg to a baby worm to an adult worm, and then a very, very old worm really quickly. So we can study, for example, the process of aging really easily using this worm. Now, as I mentioned, the ones in your garden aren't going to be glowing green like this. So in order to make it glow like this, scientists need to introduce what's called a fluorescent protein. And next up, you're going to hear a little bit about why we use fluorescent proteins. Okay, go ahead to the next slide, please.
Excellent. And as you can see, those are very, very cute pictures. But the question becomes, why do we want to have colorful worms in the labs? And what's the point of that? And basically the answer is that if we can mark something with a color, we can actually see it and understand it better. So as a question for you guys, does anyone remember the difference between an organ and a cell? And I'll just wait for a couple of minutes in the group chat and see if we have any answers for that. Exactly. So Sarah answered this question, and it's an organ is made up of many cells, and that's the correct answer. So an organ could be something like our heart or our brain, and there are different types of cells within the organ that makes them up. So with those fluorescent proteins or colorful proteins, we can visualize first an organ or a tissue, and we can visualize, let's say, a brain, and then doing different experiments, we can see if any of those experiments make a dysfunctional or an abnormal structure. So in panel A in those slides, we see some zebrafish, and we have used those fluorescent proteins to mark the brain and the spinal cord of these proteins. And this is very important for the reasons that we mentioned. We might apply a lot of treatments, we might make genetic alterations, and we want to see if any of them result in brain structures being different than normal. But at the same time, sometimes scientists are not really only interested in what happens at an organ or a tissue level, but we're interested to see what happens at the cell level. And the cool thing about those colorful proteins is that we can use more than one of them at once. So as you see in panel B, we see again some zebrafish, but this time we have marked many different cells within the brain and the spinal cord. And this is very important because we can address questions like, are those cells dividing? And how are the cell divisions changes through time? What happens to these cells, to every single cell within the brain and the spinal cord over time? So these are all very cool questions. And we can move to our next slide to kind of give you an idea of what scientists encounter when we check fluorescent worms under the microscope. So here you can see some section of worms expressing fluorescent proteins at different organs and tissues, just to give you an understanding of what this looks like in real time. And we can also move to the next slide. We are now wondering if you're ready to be a scientist, and we really need your help answer some questions that we have prepared for you. And you can answer those questions by voting in either the chat or using the polls that we have prepared for you.
All right, so I'll just let the poll run on for a little bit. Okay, could the SRHack please close the poll and bring up the results? Okay, so the question here was whether or not these cells, which are actually called HeLa cells, which come from human beings, whether or not there's any difference between these cells. And it looks like a lot of you answered C, both, which is the correct answer. So the presence and shape, the presence of dots and the shape of these cells are actually different because in B, this is an example of a human cell that is under stress. And when our cells are under stress, certain things can happen. One of them being the formation of these little dots here, which are called stress granules, and the other being a sort of adaptation of the cell into sort of a different shape depending on exactly what kind of environment has been subjected. All right, so could we please move on to the next question? So this next set of images are from an animal that scientists refer to as Drosophila, but you might know as the common fruit fly. So Drosophila is one of the more common types of model organisms, and here are two pictures of the fruit fly wing, but clearly they don't really look the same. So something here must have gone wrong. Could the rep please pull up the poll for the second question? Right. So the question here is which of these wings are normal, and in biology we also refer to the normal appearance as wild type. So just wait a little and see which one people think is the normal appearance. Okay, so it looks like most people have gotten, have chosen A as the correct answer. And that is in fact the right answer. A is the normal or the wild type fruit fly wing. You can kind of tell if you've seen insects up close, maybe under a microscope. They have very thin wings, and obviously fruit fly B doesn't really have thin wings. So this is actually a result of a mutation in fly B that has caused issues with how the wing unfolds, and it sort of messes up as it grows into a full adult fly. So next slide, please. Okay, so here we're looking at a mouse that has either one color in slide A or many different colors in image B. So you might see the word transgenic on the slide. So this is just a fancy word that scientists use for an animal that might be using genes from a different organism. So could the rep please pull up the poll for number 3? Thank you. So our question for you is, can you think of reasons why you might want to use more than one color? And our options are A, because they look nicer, B, because they let us zoom into cell behaviors, and C, because they allow us to trace cells through time, and D, because they can prove that science can be cool. Okay, so a lot of answers that are all sort of hitting the correct answer. So I'm willing to accept D, but I guess the most correct answers would be B and C, because they let us zoom into cell behaviors and because they allow us to trace cells through time. And if you have any of your own answers that you might think could be a correct question, you could answer that in the chat. So the correct answers are B and C. You can use different colors to see how different cells might change over time and whether or not they can interact or change shape. So these mice with all of their different colors are actually called confetti mice, and they're used in a lot of different scientific research. So really interesting kind of model organism there. Next slide, please.
Okay, hi everybody. So for question 4, we have some very cute kitties right here, and we want to know what you guys think. Which of these cats are not wild type or not normal? So would the presenter be able to show up the results, please, the answer? And I'll give you 1 minute to answer. Okay, so we have almost half who answered, so this could be a bit more time to put it in. All right, so 1 minute has passed. So the correct answer is A, right? The green cats right here. And so what's happening with these cats is that they essentially have a fluorescent protein, as you might have heard earlier, attached to an antiviral protein. And this would theoretically help the cats fight off any viral infection. So the reasons why a researcher would want to attach these two proteins together is because they want to make sure that the attached protein goes correctly into the right tissues or cells. And so basically the fluorescence will help the scientists determine for sure that it's in the correct place. So let's go to the next slide. All right, so over here in question 5, we have these two baby zebrafish, and your job is to figure out what's different about the one on the right. And the orange colors is for the blood vessels. How's it like? All right, I hope that was enough time for everybody. So you can now guess your answers right here. So 20 more seconds. All right, so I'll give a bit more time just to make sure everyone's gotten their answers. It's okay to get it wrong. We'll explain later. Okay, now, so The correct answer is actually D. The fish is actually flipped over, and we're looking at its belly side on the right. So I'll just show it again right here. So the orange is supposed to represent the blood vessel, and if you see closely on the right, we could see that you can see slightly growing on the back right there. So I hope everyone enjoyed that video. And now we shall go on to the next slide.
In this slide, we're looking at a baby zebrafish again, but this time we're just zoomed in on a specific region of the fish. So I hope you can see we've got a few different types of structures that are visible in this video here. So the question is, what is that big bright stripe in the middle? And yes, great, we've got the poll. And while you guys think about that, I see that we've got a question in the Q&A, where do fluorescent proteins come from? That is a super great question. And if any one of my fellow panelists want to jump in and add to my answer, that would be great. But so my recollection is that the first fluorescent protein that started being used in these types of experiments actually came from jellyfish. And that protein is called green fluorescent protein, or GFP. So that kind of makes sense if you think about all of the weird glowing creatures that you can sometimes see on nature documentaries when they are talking about the deep ocean. A lot of those animals are adapted to produce proteins that can give off light, which is really useful when you're in the deep ocean and everything is really, really dark. Since that time, we have a lot more fluorescent proteins. Some of them come from specific adaptations that we as scientists have made to green fluorescent protein or other fluorescent proteins that are found in nature. Another one that's not fluorescent, but actually bioluminescent comes from fireflies. So if you ever hear about firefly luciferase, that's another glowing protein that sometimes we use. Anyone have something that they want to add to that before we go ahead with the answer?
I think your answer was amazing. Pretty good overview.
Phew, okay. Great, so we have some answers from the poll. We were asking, are they one huge cell, a bunch of cells stuck together, a muscle, or a nerve? And you guys are exactly right. It is a bunch of cells stuck together, and that is a nerve. So what's shown here is the spinal cord in this zebrafish. All right, could we head to the next slide, please? Okay, great, thank you. Now we're showing the spinal cord of the zebrafish again, but this time there has been an injury to the spinal cord that's shown with this white arrow here in the middle. Now this injury happened on, well, here on the left, you're showing what happened one day after the injury. And then here on the right for panel B is what the fish looks like on day 3 after the injury. So our question is, what happens to the fish after the injury? And could we see the poll for this question, please? Perfect, thank you. So does nothing happen? Does the injury heal? Does the spinal cord reconnect? Or does the spinal cord disappear? And you can see the spinal cord here in the green. Okay, you are all absolutely right. The injury heals and the spinal cord reconnects in these zebrafish. This makes zebrafish a really interesting model to study spinal cord regeneration because, as many of you probably already know, humans can't regenerate their spines this way if we suffer a spine injury. So researchers can use zebrafish to look at this process. How does the spinal cord manage to reconnect? And hopefully we can use that understanding to try to figure out how we can make our spinal cord injuries heal better as well. Does anyone have any questions about fluorescent proteins or about specifically spinal cord regeneration or about zebrafish?
The next slide.
Sure.
All right, everybody. So now is your time to ask us any questions or if you want us to repeat any videos, please let us know. And so just type in the chat, right? Or in the question and A poll. Okay, so I see we have one open right here. So can you show these samples under the microscope? So if anyone wants to add pitch in, please go ahead.
Sorry, could you say that again? Add what?
So does anyone want to answer first or should I?
Yeah, go ahead.
Okay, so basically, yeah. So a lot of the samples you've seen in this presentation, they're basically under the microscope. So we use these specialized microscopes to view tissues, and these fluorescent proteins help us visualize these cells or these tissues with these bright colors. I hope that answers your question, Tanya.
I think there might have been one or two out of all of these that are not under the microscope. Can anyone remember and guess which ones? I'm not sure if you have a way to answer that. Yes, the chat.
In the chat? Okay, I'll check there.
Cat, yeah. Yes, exactly. Yeah, the cat's big enough to see.
Yeah, the cat, you don't need to view it under a microscope, thankfully. But for like smaller animals, such as the worms or the flies, and to a lesser degree, even the mice, if you're bringing them to your embryonic state, like when it's still developing, then yes, you can see them under a microscope.
I see there's a question in the chat. Do you mind if I take that one?
Yeah, sure. Go ahead.
So this question is, why are you using this cool animal model, zebrafish, compared to other models, e.g. mouse, to study spinal cord injury? That's a fantastic question. And I might have seen you on my grant committee. The main reason that we're using zebrafish instead of mice in this case is because mice are more like us in the sense that they don't regenerate their spinal cords that well. Mice and rats and a lot of mammals are actually not fantastic at regenerating compared to zebrafish. Zebrafish actually, they can repair their spinal cord and reconnect it a lot better. So if we want to ask the question, how does regeneration work? We want to use a model that can do it really well. So that's the number one reason. The number two reason is that zebrafish are really fantastic when it comes to using fluorescent proteins. When they're in their larval stages, so as they're developing, they're totally clear. So like you saw in the developing zebrafish video, you can see right through them. So it's really, really easy to image them under the microscope and really clearly see those fluorescent structures. That's why we can get such nice videos and images from them. So they're a really great model when you want to use fluorescent proteins to examine a process. So those are the two reasons why zebrafish is what we study in my supervisor's lab rather than mice. A really great question. All these are great questions. Yay, go fluorescence protein and go basic science research.
Those are the best. All right, so I'll check the questions answered again. Okay. So anyone else have any other quick questions, or would you like us to show you any other the older videos just to see how pretty they are? in the meantime, so would everyone like to go over which labs they work on, what kind of models they use?
I can go first if no one else wants to.
You work with mice, right?
I do. Unfortunately, not glow in the dark mice. I just work with normal mice. So I do microbiome research. So this is usually with actually from samples that come from people, as well as samples that come from mice. So one of the things I will do is take samples from the mouse gut microbiome, which actually comes from their poop. So I wait for them to poop, collect it in sort of a little tube, and then I check it for DNA and something called RNA. And I check whether or not certain mice have different kinds of RNA or different levels of RNA, depending on whether or not I, for example, feed it this one food. So you'd hope that perhaps those mice, their microbiomes, which is a collection of bacteria in their guts, perhaps they respond differently depending on what you feed them. So we have another question. So where do you get those zebrafish and mice from? If someone knows the answer to that, I can try to give my answer, but I don't work with zebrafish.
I could answer that, but so the amazing thing with all these model organisms is that it wasn't really a mastermind behind them that was kind of thinking, oh, what model organism could I use? Most of them were identified very randomly. Like you can see, for example, that yeast cells, they do divide randomly, and they do divide nicely, and they do have specific morphologies in different stages. So people who observe them are like, maybe I can use those to study cell division because it's very distinct. And the same thing came from zebrafish and mice as well. Mice are very, very similar to humans, and they're a great system to study diseases. As for zebrafish, as Nicole mentioned before, you can see that the embryos are see-through under the water. Also, the fish leave their embryos free air, so you can have access to them and you can visualize them. And that's how people decided to use those to study specific things. And now it's a bit boring of where we get them from. There are companies that they do go do these mice breedings, and very, very very well used strains of these. They're available through companies that you can order them. And they're labs that they do make their own strains. So they're labs that they want to change some genes in these organisms to make them more suitable to study different diseases or different processes. And that's could be lab to lab specific. And usually those ones are not, you can order them again, but usually the labs like to keep them and they focus the research on them.
We have a related question about how much would these mice or models cost? I believe it depends on a company, if I'm not wrong. And what kind of mice strain you're looking for, like the type of mutation it has in the DNA.
Did you know though that you can buy glowing zebrafish at PetSmart? I think those ones cost like $7 a fish. I'm not. I know that there was some concern at one point that the fish would accidentally be released into the ecosystem and could cause problems. So if you do get one, I guess it's good to be very careful and not dump it down the drain. But those ones are actually available for purchase. But oh, sorry, go ahead.
I'm sorry to say that the glow-in-the-dark cats are not available. Why
It's probably for the best. I mean, cats are already like quite active at night and like making a lot of noise and stuff. And imagine a glow-in-the-dark one like waking you up inches from your face.
I don't even think about that. So yeah, in terms of cost, I think it does vary on the company strain. I hope that answers your question, Sarah.
We have a question in the chat. Does the zebrafish feel or know that you are there? I think that's a really good question. For cats, if we're talking about cats, you know that they do know that you're there. For fish, I think it's a little bit more of an open question as to how much they know that you're there or that you can interact with them. Sometimes they will run away from you when you're trying to scoop them up with your net. But for all of these animals, if we're working with them, we have to be very careful about how they're feeling when we're doing these experiments. So we have very specific protocols that we need to follow to make sure that they're not going to be uncomfortable or in pain from the experiments that we're doing, and that they're really well taken care for and or taken care of, sorry. And that applies for zebrafish and for mice and for cats. We always have to make sure that that they're living good lives and that we're not causing them pain from the experiments that we're doing.
I should want to add, though, also added that there are specialized attendants or assistants for these animals as well as veterinarians to make sure that they're in good health and state. Does anyone else have any more? Okay, Sarah has another question about, do you work on monkeys? And I would also have a quick question, answer of mine that, yeah, some researchers actually do research on monkeys. If anyone else want to add to that, or?
So I work with an organism that used to be studied in monkeys. It's a certain bacteria that I'm sure not many people know of, but it used to be worked within chimpanzees, I believe. And because the bacteria is human specific, chimpanzees were the closest thing that they could find. And not a lot of scientists work with chimpanzees anymore, and monkeys in general, just because of really the cost and also of sort of the concerns over the health and safety of monkeys, as they're a lot harder to keep happy than something like a mouse or a zebrafish. So that's, I haven't worked with monkeys, but it's definitely a lot more rare than you would think.
Mm-hmm.
Although not me personally, but some of my friends do work on brain cells that are taken from monkeys that they then are able to grow into what's called an organoid. An organoid is a structure made from cells that is kind of like an organ, but isn't quite there all the way. So it's basically like kind of like a monkey brain in a dish, but it's not quite all the way to being a full brain. So it can't really think, but the cells act in the way that they do, or some of the ways that they do in an actual brain. It's kind of a way of working on a monkey without really working on a monkey.
Yeah, I would agree.
Why we might not prefer monkeys and cats on our experiments, and we might just want to work with some other organisms like fish and mice.
Yeah, I would agree with that. I would like to add that working with mammals in general, like cats or monkeys, it's quite much more rare than it used to be. And there is primate research in the US, for example, and perhaps some other research institutions in Canada. But in U of T, I don't believe we have any monkey research specifically. Someone can correct me on that if I'm wrong.
I think oh, sorry.
Oh, we have another question in the chat, which is, do you share with other scientists around the world your animal models? And if so, how are they transported?
Ooh. Correct me if I'm wrong, but I believe that since a lot of these models are commercially available, that multiple institutions are able to get them. And if the scientist chooses to, they can, I believe they could.
We definitely share our zebrafish with other scientists. If you make a type of zebrafish that has a fluorescent protein in it, you might want to share that with other people, and they can be transported with great care. And sometimes they get stuck at customs for a while, and you have to rescue them. Yeah, if it glows at the customs, then it might give somebody a real shock. Definitely, I think that flies are easier to transport and worms compared to fish or mice.
I had to send some mice to another lab at some point, actually, and all the bureaucracy and all the paperwork you have to fill is a nightmare.
Yeah, you have to be super careful to make sure that like the new organism that you're bringing in isn't carrying any diseases or anything that's going to harm your existing organisms.
Yeah, I believe that in general, it could be quite tricky bringing living things or across the border in general. That counts not just for model organisms, but even produce, for example, would be tough. All right.
I want to say thank you for all your great questions, everybody.
Yeah, these are great.
Yes, I'm recruiting you all for my lab.
Your PI would appreciate that for sure. Okay.
So waiting for questions, do you want to tell us more about your project, Nicole?
Oh, sure. So I've been lying to you all. My supervisor's lab does work on zebrafish, but I don't work on zebrafish actually that much myself. I actually work on a type of worms that we didn't show in this presentation. They're flatworms, and they're really cool because you can take one flatworm and chop it up into 10 different pieces, and then each piece will grow back and become a whole new worm. So I work on also the question of how does this happen? How does it manage to regenerate like this? And we have a much harder time making those worms glow because they don't work the same way with regards to expressing fluorescent proteins. So you can do it really easily with zebrafish, not super easily with flatworms. But they are really cool animals to study. And if you're curious about that, definitely let me know, and I'm happy to talk about it. We also have a question, what are some downsides for working with a fluorescent microscope? I'm sure you guys are feeling very familiar with those. Anyone want to take that?
Well, most fluorescent microscopes, they can serve as normal microscopes too. So it's not that you only use them to see fluorescent proteins. You can change the channels and you can just take nice pictures. So just use it as a regular microscope. But downsides for working with them, with fluorescent microscopes, other than the practical things of being expensive. Well, something that people need to keep in mind is we show you all these great pictures of model organisms that we use many colors. So sometimes one color interferes with another. And if you want to use more than one, you might need to be a bit careful with what combinations you're using. The downside of working with some microscopes is also that well, different microscopes have different capacities. So depending on how many proteins you want to see, what are the different fluorescent proteins you want to see, you also need to be careful choosing those. Does anyone want to add to that?
I think I may. I think the process in general of creating fluorescent organisms can be quite challenging because you have to code it into the DNA of the organism, insert the genes. So one could argue that could be a challenge as well. Oh, there's another one. Or I guess if someone else wants to add as well. If you transplant a green mice organ into a normal one, will it still glow green? As I mentioned before, I also answer a quick question. So basically, you need to insert his DNA first before it can glow. So I'm not too. Maybe the organism itself won't glow, but there's a chance it can pass off to its offspring. Someone else want to add to that?
I'm doing that a lot. I'm transplanting. I work with cancers and tumors, so sometimes I do create those tumors that they do express TFP, so they're green, and I transplant them in other mice. And we want to see if those tumors do grow more. So one way to check if it's actually your original tumor that grew, or if it's a side effect, that if your new mice just had a tumor on their own, you just isolate this tumor again and you can check it under the microscope to make sure that it is TFP. So if you have an original organ that is genetically modified to express this proteins, then you transplant it. In different mice, yes, they do usually maintain this. But it depends on the mice strains. Usually what you want to do if you're transplanting organs or tissues or cells or anything, sometimes you want to move them to immunocompromised mice. So you make sure that their immune system is not going to fight against the new organ you're introducing. So that's also something to keep in mind.
I want to say that's such a great question.
Yeah. I think all of the questions we got are great.
Mm-hmm.
You guys are way smarter than me, so
Lots of good scientific thinking, for sure.
I do want to thank you, Sophia, for correcting me on that one, to be honest.
Oh, no, it was more like adding. I don't think I corrected you.
All right. So I also just want to mention that the organisms that Nicole was talking about, I posted in the chat. So if anyone wants to Google them, look at videos, I would highly recommend it because they're actually quite cool. Let's just wait for a few more questions.
So what do you work on with your research?
So I'm actually a staff member of the Department of Molecular Genetics. I work as a science writer, so I basically work with the communications part of the admin. So unfortunately, I don't have any cool stories, research projects to mention to you all, but hopefully in the future. So basically, I write stories about other people's research, but I am not in the lab myself.
Can you give us your opinion on which model organism you would most like to work with?
That's a tough question, honestly.
Planarians.
Yeah. Yeah, I guess zebrafish would be right. Zebrafish or planarian would be the best. Oh, I think this would be a better question for the rest of you. Any fun stories about working with these animals? I feel like Ryan would have a tough time with the mice sometimes.
Yes.
Yeah, maybe not a fun story, I guess, for myself, but it's always funny when a lab member will come up and they've been bitten by their mouse and they swear off doing mouse research. It's always a fun time. But my mice are really nice to me, so that's not haven't happened to me yet.
That's great.
The other fun fact about mice is that not only they bite you, but they also pee on you.
Oh, do they poop on you too?
Yes. I think for me, the poop is like fine, but as long as it's pee, I'm like, okay, whatever.
Because the poop has science value in research as well.
Yeah, in my case, I want them to poop on me.
to hear.
For me, thank God it doesn't. So I don't have to collect it.
Yeah, I can imagine.
Kiki says, fruit fly scientists like to push flies around while listening to Beethoven. It's also true for cell culture people. When I did cell culture, I always sang to my cells while I was alone in the tissue culture room. So that's maybe a next level of that.
Yeah, and also I can imagine trying to catch the flies and making sure they don't fly out the room as well, or their cage.
Fun story. When I was in undergrad, we had a class that was doing experiments with fruit flies, like very basic experiments. And one of the things they teach you, like you can keep your flies in those vials. And one of the things they teach you is how to transfer flies from one vial to another.
Okay.
And because it's the first time that they teach you how to do that, and if I remember that correctly, and if there's a Drosophila person, correct me, like you tap your vial on the bench and then your fruit flies go down and then you quickly move them to another one and you close it. But that being said, being the first time you do that, it's not quite efficient. So I do remember that when we would open the door to exit the lab, it would be a swarm of flies also like exiting with us. So that's my fun story of dealing with flies.
Oh, geez. You see, I think this is why zebrafish and planaria flatworms can be preferable, I guess, because
Although
they don't fly around.
I can definitely add that for the zebrafish, when you are holding them in the net, they do sometimes jump out. So you have to be very careful. And if the fish lands on the floor, you can't put it back into the tank with the others because there's a concern that it may have picked up some germs on the floor. So if you have a fish, you can't let it touch the floor. And if it jumps out of the net, you might find yourself going, trying to juggle the fish and catch it before it goes on the floor.
Yeah, it's the same with mice. Like if they fall off the cage, and they fall on the floor or something, unfortunately, you cannot just put them back.
Wow, that's cute. I love how you guys were sharing your stories about working in the lab. I think that really gives, like, especially all of our attendees, a quick idea about the fun parts of it, the hard parts of it, but really how rewarding science really is. So thank you all for sharing and for joining us today. It was so lovely getting to also see the cool parts of science, right? The pretty pictures. Super nice, super fun. And yeah, okay. So a big shout out to the entire molecular genetics group. All 3 presentations were incredible. Thank you to Kiki for helping organize everything as well and for staying on. And yeah, so if there are any additional questions, we can definitely leave the Q&A open for everyone. And if you are looking for any additional information, I'm sure you can go up and search the molecular genetics on Google under U of T and find some more information. All right, so jumping right into our next group, we have the David A. Dunlap Department of Astronomy and Astrophysics. So we're moving in from the really tiny little cellular molecular aspect, and we're moving to the complete other end of the outer space. So that's a cool transition, but again, so right now we'll be exploring the sun with the team from Astronomy and Astrophysics. And I do want to remind all the attendees once again, I know I've spoken like a broken record, but do post your questions in the Q&A. It's super exciting getting to really have the chance to interact with the attendees and the speakers. And yeah, without further ado, let me give our Astronomy and Astrophysics team the floor, and super exciting.
All right. Hi, everyone. Welcome to our Astronomy and Astrophysics segment of Science Rendezvous. We're really excited to see you all here. My name is Ilana McDonald. I work in the Astronomy department at U of T, the David A. Dunlap Department for Astronomy and Astrophysics, if you want the full name. And we've got several exciting things for you coming up today. So we're going to start with Adeze Ibik, who's going to she's a PhD candidate in the department here, and she's going to talk to us about the sun, give us some background information, what it's made of, how it works, and a lot of cool stuff about it. Then we are going to have 3 trivia segments in this segment, and these will be run by Nicole Gromick, who is a recent graduate of the astronomy program. And then we are also going to have some live solar observing. So our local telescope guru, Mike Williams, who's a staff member in the Department of Astronomy and Astrophysics, he's going to be showing us some live views of the sun. And we're particularly lucky today because it is gorgeous and sunny out. And then the final thing we're going to show you is a virtual observatory tour. So even though you can't visit our observatory in person, you can at least see what it's like. So we're going to start with Adeze, who's going to talk to us all about the sun.
Hi, everyone, and hopefully you can hear me, right? Good. Yeah, so I'll just go ahead and share my screen. Yeah, so today we'll be discussing a little bit into the basics of the sun. So the image that you can see right here is a recent image of the sun just last month, which is interesting. You know, like you could see so many interesting features already. The fact that the sun isn't even white is one of the, very first thing you just see right away. So what is the sun? I would like people to think about what the sun is just with your senses. Can you see the sun? I think everyone can. Can you hear the sun? Can you feel it? Can you smell it? So one important thing is that everyone feels the hotness, just like everyone is expecting summer very soon, and we begin to feel the scorching hotness of the sun. And then it's very bright. In fact, from far away, it looks like it's white in color. So we are going to be digging into what is really the sun. Like I've shared in the first slide, is not really white, but of course, because of the atmosphere and then because it's far away, we end up seeing it as if it's white. So getting into the scale of the sun, how big it is, we know it's a very large object. We try to understand the mass of the sun when we look at the planets. The planets in our solar system, they all revolve around the sun. And so we learned that the sun is so massive in terms of mass when we understand the orbits of these planets. And we also understand that it's big. It's not just massive, but it's also very big. So when Venus or Mercury is passing in between the Earth and the sun, which we call the transit, we are able to use that to understand how massive the sun is. So when you look at the image on the side, you notice that Venus is able to pass through the sun, and we're able to capture that, that frame. And that is what we call the transit of Venus. Now, this shows you how small Venus is compared to the sun. So it turns out that we could have like 1.3 million Earth fit into the sun. That tells us how large it is. So the sun ends up making up like 99.8% of the entire solar system. So every other planet that we know and every other object in our solar system make up less than 1%. So the sun is mind-blowing, isn't it? And so we want to understand where the heat from the sun is coming from. What is powering the sun? What's fueling the sun? Could it be, like the way we heat up our houses during winter in Canada? There are different natural gases that we burn to produce the heat in our houses just so that we can feel comfortable. Could that be what's powering the sun? So for us to understand how the heating of our houses works, so we need to understand what is a chemical reaction. So the natural gases born in a process called a chemical reaction. Now, the chemical reaction, happens because atoms combine to form molecules. Now, atom is a new word that is made up of protons, neutron, and electron. So these things combine in different forms to make up the molecules. Now, there are molecules like oxygen and also atom like oxygen. We have carbon also as an atom, but when they combine different atoms, you get molecules. And so for the chemical reaction that heats up our houses, I'm going to use an example of a natural gas called methane. So what you're seeing here is like methane combining with oxygen molecule, which now produces water, and the carbon dioxide, which is what now produces the heat we experience. So we've tried to understand if this could be what's powering the sun. Now, it turns out that for this chemical reaction, though all of the atoms remains the same after the reaction, nothing is destroyed, nothing is created. So the heat it's producing is not able to produce the heat we experience from the sun. So what then is powering the sun? Since this is not enough, it turns out that something called nuclear fission is powering the sun. So what is nuclear fission? So hydrogen atom, combine into different, under different conditions to form helium. And so this process ends up having a lesser mass as products, which mass is now converted into energy using the famous S.T.E.A.M equation, like you can see on the screen. So this is how we end up powering the sun. How does it happen in real life? So when the pressure and temperature around the center of the sun begins, to have the right amount of, value, like when they have certain conditions become so hot and so high, then it's able to force the hydrogen atoms around to begin to combine to form helium and other particles. And so this process produces enormous amount of energy that's able to power the sun. And in fact, the sun ends up losing like 3 million atoms every second. But then it's like it's tiny bit because the sun is huge. So now that we understand what is powering the sun, let us see, what is the sun made of? So we now know that the sun is made up of hydrogen burning to form helium and other particles. So when we look into the sun, we actually see that it's also in like layers. There are different layers of the sun. Just like you can see from the image that I'm showing, you have the central region, which is the core, where this nuclear fission happens. Then you see that coming towards the surface, you have the radiative zone, where the energy, the light produced at the core is beginning to transmit towards the surface. Now, the movement of this energy takes it over to the convective zone, and there you begin to experience what we call convection. So there are movements of plasmas and gases. So plasma is streams of particles, and gases are all the gases you can think about that are produced during this nuclear fission. And so this process is also very evident, such like this, the diagram by the side showing, like what the convection causes. It makes the region look like granules, like you see the very dark region where it's much colder, which I'm going to talk about in a bit, called the sunspot, and also the lighter region where it's much hotter. So when you keep going outward, as you go outward, it gets colder. From the core, the temperature is like, 13 million degrees Celsius. And as you go out, it gets colder. And then when you get to the very surface, which is called the photosphere, the temperature is really, really cold. So 5,500 degrees Celsius is really cold. So we have this temperature for the surface of the sun. But then when you go out a little bit, which we call the corona, something that looks like atmosphere of the sun, that ends up becoming very high again in temperature. So the corona seems to be quite high, even though it's like the atmosphere of the sun. So it makes it interesting to understand what is heating the corona. So it's about 12 million degrees Celsius, which is insane. So this is one of the things that's puzzling about the sun, that we don't understand what's heating up the corona. Now, one other feature I would like to talk about is the sunspots, which I've shown here. So sometimes we see these sunspots, some dark patches in the sun, and those are areas of, high magnetic field of the sun. And those look really, really dark just because they are colder. So the magnetic field traps the cold gases in those areas so that they don't get to sink back. So during convection, hot gases get to the top while cold ones sink back down. And so when they get hot again, they come up and the cold one goes down. So you just continue that process. But when the magnetic field is strong in a region, then it doesn't allow the cold one to sink back. And so we're able to see those as sunspots. There is also the solar prominence, as you can see in the image. This prominence is the plasma that are like very big, like giant plasma coming up, and ending up on the other side of the sun, which is really huge. So these are some of the things you probably could see today when we'll be showing you some of the observations, hopefully. So one thing to note about the corona that I mentioned earlier is that we are not able to see the corona with our naked eye. So most times we are only able to see that during the solar eclipse. So when you have a total solar eclipse, the entire photosphere is covered, and then it allows us to see the corona. Just like you can see in this other diagram. Now, this is a typical eclipse, and we are able to see the corona as those whitish particles. Now, this is very hot. Now, that reminds me to mention that we'll be having the closest solar eclipse in 2024. So that will be another total solar eclipse for Ontario. Maybe not in Toronto, but like places in Hamilton, Niagara Falls, I think, will be having a full solar eclipse. So that's something to watch out for, and we'll be able to see the corona whenever we have these eclipses. So I mentioned that the corona have been interesting. So scientists think it's what, understanding how that area is heated up. And so recently in 2018, a satellite was launched called the Parker Solar Probe. The instrument you are seeing here is a very powerful instrument that was launched recently to study the heating of the corona and, of course, understand the solar wind. So I forgot to mention that the sun always emits streams of particles in form of solar winds into the solar system. And some of them end up getting into Earth, and we see them as the northern lights. So sometimes you see the northern lights. These are, caused by the solar wind. So I will be talking a little bit about the Parker, just to help us understand how much we've learned about the sun. So recently in 2021, the Parker telescope have sent some of the videos and pictures of the corona, which I'll be showing in a bit. But one thing to keep in mind is that because the corona is quite hot, this instrument has been built such that it can endure the hotness of the area it's going to be visiting. And so that's why it's able to work. It has a camera that is able to capture the scene, like it's able to see the solar wind, it's able to see some patches of the sun. It's also able to measure the magnetic field, through the sunspot region, and some other things that it's doing and sending us data. Now it's having an elliptical orbit around the sun, and really passes through Venus while it's making that movement. So I will just play this video just so that you understand what we've been looking at. So you can see the string, one of the arms of the Milky Way, and the streaks of particles. These are solar winds that are crossing the line of sight of the Parker Solar Probe. You can see Venus. This planet is Venus. You see the Milky Way. This is just one of the spiral arms. You can see some other objects around. And of course, the streaks are the solar winds. really looking quite fast. These are streams of particles, coming out from the corona all the way to the Earth and other parts of the solar system as well. Okay, I will stop that. So this is coming from the solar probe. And it's interesting that we could see, very close to the sun. This is the longest, the farthest we have gone to the sun, and it's a very interesting instrument because it's also moving fast. It's the fastest thing we have also made, scientists have made so far. It's able to move like as if you're going from Toronto to Ottawa in 1 second. That is how fast the telescope is. So, which is incredible, right? So, wrapping up, just to mention that while it's interesting for us to look at the sun, please do not look at it with your eyes. Do not stare at the sun. It's dangerous. You must look at it using instruments with proper solar filters. And also you can project the sun into a camera or through a camera into a paper or something like cardboard or using a pinhole. Also, just like you can see in the image, and you're able to see the sun that way. You can also spot the sunspots that way. So it's just safe that you don't look at the sun directly. It can damage your eye. I hope this is helpful, and we'll be going over to the trivia and some other interesting things that we have for you.
Thank you so much, Adeze. So now we're going to be going over to Nicole, who will be running our first trivia segment.
There we go. Welcome everyone to our first trivia segment, where we will be testing your knowledge of all things sun-related for a chance to win some prizes. We will get started soon, so please go to kahoot.it and enter the code to join. While we wait for people to join, I'll do a quick rundown of what to expect. There will be 4 questions per segment. We have about 20 seconds to answer each question, and speed also matters here, so answer the question faster to get more points. If you want prizes, you're going to have to put your real email address. We promise we will only send you an email if you win something, otherwise you won't hear from us. And you can only win 1 prize today, just to keep things fair, but you can absolutely keep playing after you win. If you're under 18 years of age, please get permission from your parent or guardian to play. And yeah, I'll just wait for people to trickle in. I think we'll start in about 30 seconds. I have to say the prizes look pretty good. We'll show you the prizes in just a second. And yes, you can feel free to join the game at any point while we're playing it. The code will be on the screen at all times. And I think with that, we can get started. So our first question is coming up. Oh no, here's the rules again. So just like I said before, you have to put your real email address. If you're in Quebec, I'm sorry, but we can't send you a prize. And you can only win 1 prize. And if you're under 18, please make sure you have your parent or guardian's permission to play. But here's the most important thing: is the prizes. First prize is we have the moon globe, which looks fantastic, and we have some sweet Dunlap merch for 2nd and 3rd place as well. And with that, we can get started. First question: how many times can the Earth fit inside the sun? Is it 1,400 times, 160,000 times, 1.3 million times, or 4.7 million times? Perfect. The correct answer is 1.3 million times. That is a lot of Earths just to give you a scale of how big the sun is. So right now, Proud Bison is in first place. Let's see if you can keep there. Next question. Approximately how old is the sun? Is it 13.7 billion years old, 6.3 billion years, 4.6 billion years, or 1.3 billion years old? I think I might need a birthday cake for it soon. Ooh, it was a split between 6.3 and 4.6, but yes, 4.6 billion years old. And fun fact, the Earth is actually not that much younger. It's just 4.5 billion years old. Let's take a look at the leaderboard. We have Fast Ibex in first place. Ooh, things are getting tense here. How long does it take light emitted from the sun to reach the Earth? Our options are that it's instant, that it takes 8 minutes, or 11 minutes, or 2 hours. Perfect. A lot of people got the right answer. That is 8 minutes. And of course, light is so what we call the speed limit of the universe. Is that the fastest thing that can move? But it still takes 8 minutes to reach us. Oh, we'll be going into our final question now. What form of electromagnetic radiation is responsible for sunburns? Is it visible light? Is it ultraviolet, infrared, or microwaves? Perfect. So the correct answer is ultraviolet, or UV rays for short. And that is just a reminder to wear sunscreen. It is very important. And it's time to see who's on the podium. In third place, we have Proud Bison. And next up in second, we have Diplomat Emo. And in first place, drum roll, we have Fast Ibex. Congratulations to our winners. We will contact you via email about the prizes. And to everyone else, please keep in mind that we have 2 more trivia segments to run, so stick around for those.
Thank you so much. That was awesome, Nicole. Next, we have Mike, who's going to be showing us some live images of the sun.
Hi, everyone. So I'm actually on campus at U of T in our observatory, which is on the 16th floor of the Planet Physics Tower. And I'm going to start out using our 8-inch telescope. So our telescope looks like this. So it's a nice big long tube, and it has a lens right up at the front, and that lens focuses the light down into a camera. Now on the very, very front of that telescope is a filter that looks like this. So it looks like a mirror. And that reflects away almost all of the light that hits it. Only about 1/1,000,000th of the light that hits the front of that filter actually gets through it and to the camera. And that's really the only safe way you can look at the sun through a telescope. Otherwise, you can burn your eyes or set fire to your camera, all sorts of things. So through that 8-inch telescope, this is what the sun looks like. And we can see a few sunspots. So they're just right here. Yeah, so sunspots are slightly colder parts of the sun. So the surface of the sun is about 5,000 degrees Celsius. And the temperature of the sunspots is about 4,000, maybe 3,500 degrees Celsius. So it's still really, really, really hot for us people, but it's slightly colder than the surface. So it looks black. So our 8-inch telescope isn't the only telescope we have that can look at the sun. We actually have this smaller telescope. It's called an H-alpha telescope. So it's this gold telescope on the side. And that lets us look at a very special view of the sun. So basically, it makes it so that we can see much, much better contrast on the surface of the sun. So right now, we're looking through the H-alpha telescope at the sun. And the first thing you notice is the sun's actually backwards the way it was before. So those sunspots we saw before, which were on the right, they're actually on the left. That's because this telescope actually has an extra prism in it that fixes the fact that telescopes make things backwards. But through this view, we can actually see the granulation on the surface of the sun, and we can see all sorts of different patterns caused by the magnetic field of the sun. And if I change the exposure of the camera, we can actually see some solar flares and prominences around the edge of the sun. So I'm going to increase the exposure. And now you notice around the edge, so right here, there's another one up here, and there's a whole bunch over here. So this is plasma coming up out of the sun, and it's trapped in the magnetic field of the sun. And each one of these prominences is probably about 3 times the size of the Earth. And so those will change over time. They'll get bigger, smaller, disappear. And as the sun rotates, the direction they're pointing will change. So that's basically what I have to show you about the sun today. So I'll pass it back to Alana.
Great, thanks, Mike. Those solar prominences, every time I'm like, wow, that's so freaking cool. So now we're gonna rejoin Nicole, who's gonna host our second trivia segment. So if you didn't win the first time, here's your chance to win this time. So here we are once again, looking at that.
Perfect, and welcome back everyone. So it's time for round 2 of solar trivia. Enter the code to join and we will get started in just a bit. And I hope that everyone's been enjoying their Science Rendezvous day so far. I've popped into some of the presentations throughout the day, and a lot of them were really, really interesting. Okay, we'll get started in just a couple of seconds. Okay, I think we can get started. Just to remind everyone, here are the rules again. Oh. Yes, there they are. So please use a real email, and then everything that we said before, only 1 prize. And then here is what is going to be at stake for those prizes. Again, I really want that moon globe. And coming up will be our first question. Tell me, is it true or false that sunspots are cooler than the rest of the sun's surface? Perfect, it is true. They are caused by the sun's magnetic field coming through the surface, but they are still very, very hot, coming in at around 4,000 degrees Celsius, you know, a little bit too hot to boil your tea on that. Let's check the scoreboard. In first place, we have Expert Hawk, closely followed by Genius Sphinx. Let's see how those numbers round out in the second question. How long does a solar cycle of sunspots last? Is it about 5 years, about 10 years, about 20 years, or about 100 years? Perfect. As I should explain that a solar cycle is a repeated pattern in the quantity and size of sunspots and prominences that we saw from Mike. And the correct answer is actually closer to 11 years. The third question we have coming up. How hot is the center of the sun? Is it 6,500 degrees Celsius, 2 million degrees Celsius, 7 million degrees Celsius, or 15 million? Ooh, that was a tricky one. It's very, very hot in there. 15 million degrees Celsius. Let's see how that shook up the leaderboard. Genius Sphinx still in the lead. Let's see if you can keep that lead in the last question coming up. What color is the sun as seen from space? Is it blue? Is it white? Is it yellow? Or is it orange? Three-way split. Wow, it's white actually. And this is a bit of a tricky question because we see it as yellow or orange from Earth because of the interference from our atmosphere, which is why we get such pretty sunsets and sunrises. But from space, it is white. All right, let's take a look at how those scores came out. In third place, we have Lovely Zebra, lovely as always. In second place, we have Genius Sphinx. And in first place, who could it be? Magic Gazelle, well done. Good work to all the participants. And remember that we still have one more trivia section coming up. So if you didn't get a chance at a prize, please come back.
Thanks so much, Nicole. And congratulations to all the winners. So the next little bit in our segment is a virtual observatory tour so that you can see what our observatory is like, even though you can't necessarily go there. Started at the beginning. To get up to the observatory, we'll have to take the elevator all the way up to the 14th floor. Now that we've taken the elevator up as high as it will go, we still have to take the stairs up another 2 floors in order to get to the observatory itself. We've created a model solar system going up the stairs, where the number of steps represent the distances between the planets. On the top floor of the building are located our 2 observatory domes. The first we'll go visit is the refractor dome, which contains our 8-inch refracting telescope. At the end of this telescope is an 8-inch diameter lens, which focuses light down the 2.5-meter tube of the telescope down to the eyepiece. It's called a refracting telescope because the lens refracts light in order to focus it. This telescope is older than the physics building. It was built in 1965 by the Goto Telescope Company in Japan. In order to see through the telescope, you must look through the eyepiece located at the back of the telescope. There are 3 eyepieces that can rotate to give different magnifications. The shortest gives the most zoomed-in view, while the longest gives the widest field view. Attached to the side of the 8-inch telescope is a small Coronado solar telescope equipped with a hydrogen alpha filter. This allows us to observe fine details on the surface of the sun. The large scope itself can be fitted with a solar filter, which makes it safe to view the sun through the telescope's lens. Remember, you should never look at the sun directly without protection, even with the naked eye. You'll notice that the telescope must be moved around by hand. This is because the only motorized component on the telescope is a tracking motor, which makes sure that the telescope follows the motion of the sky.
Otherwise, the telescope is operated completely manually. To allow us to view the sky, the telescope dome opens up in a 1-meter wide window that extends to the top of the dome. The dome itself can rotate 360 degrees, which gives us a complete view of the entire sky. As you can imagine, lining the telescope up with the sun can be quite tricky because the sun is so bright. The mount of the telescope is an equatorial mount, which means that one axis points towards the north celestial pole, that is, the north pole of the sky. This is parallel to the Earth's rotation axis, which means that you can easily keep the telescope pointing at an object in the sky with a single motor. Some of the best things to observe through our telescopes are the planets that you can see in the night sky. We're going to start with the planet Mars. You may have noticed that Mars is bouncing around a little bit. This shimmering effect is due to turbulence in the Earth's own atmosphere. You might have noticed this white spot at the top of the red planet. That is, in fact, Mars's polar ice cap, which contains frozen CO2 and frozen water. The largest planet in our solar system, and probably the most easy to see through our 8-inch telescope, is the planet Jupiter. In this view, we can see cloud bands, which represent weather patterns seen in Jupiter's upper atmosphere. In this more zoomed out and longer exposure view of Jupiter, you can see these 4 little faint dots surrounding the gas giant. These are Jupiter's Galilean moons, called that because they were discovered by Galileo when he first looked at Jupiter with his telescope. Another planet that we can observe with our telescopes is Saturn, famous for its beautiful set of rings. In this view, we can clearly see the gap between the rings of Saturn and the planet itself. Though it's much more difficult to observe with the naked eye, we can also observe the planet Uranus with our telescopes. Seen here is a faint green blob. The planet Uranus is the 7th planet from the Sun and is also 20 times farther from the Sun than we are. In our second observatory dome, we have our 16-inch reflecting telescope. It's called that because it has a 16-inch diameter mirror at its base. Unlike the 8-inch telescope, this telescope is fully motorized, which means that we can control it remotely. Just as with the 8-inch telescope dome, this dome can also rotate a full 360 degrees, which gives us a complete view of the sky. The 16-inch mirror is used to focus light from the sky down the tube of this telescope. This telescope collects 4 times as much light as the 8-inch telescope because it's twice as wide. As well as being used for our public outreach events, this telescope is used for research purposes by undergraduate students in their courses, as well as by graduate students to test their instruments. On the back of the telescope, there are several instruments to facilitate these activities, including a couple of CCD cameras and a spectrograph used to split light into its separate components. Just like the 8-inch telescope, the 16-inch reflector was built in the late 1960s by a company called Böhler and Chivens. Seen here are the original readout dials, which show the telescope's position in terms of right ascension and declination. Both the motion of the telescope and the telescope dome can be controlled by this computer that is located in the control room right next to the telescope. Since these telescopes have been in operation since the late 1960s, we can see some of the earliest photos taken with this telescope. Some of them are on display in the 16-inch telescope dome. You can see here several deep sky objects, including nebulae and star clusters. The photos were taken using photographic plates, since this was long before digital cameras existed. Some of the exposure times on these photos are several minutes to an hour long. This is because it took that much time to collect enough light from these faint objects to be able to see them in nice detail. All of these photos were taken by John Cormandy, who was an undergraduate student at the time, and James Gillespie, a former master's student at the David Dunlap Observatory. One floor down from the telescope domes is the observation balcony. Here we have an almost unobstructed view of the southern sky. We use this balcony to set up smaller portable telescopes during our public events. Here is that same view at night. There certainly is a lot of light pollution from those city buildings, but that doesn't prevent us from observing beautiful planets, stars, and nebulae. To finish up our virtual tour, I'll leave you with this view of the Big Dipper as seen from the roof of the physics building just outside of the observatory domes. I hope that one day you get to visit us in person. All right, and now we'll go back to Nicole for our final trivia segment. Let me just share that. So be sure to put this code into Kahoot.
Well, it is time for our final trivia of the day. I can't believe it went by so fast. You folks know the drill. Enter the code and your real email address if you want those prizes. And just before we start, I just want to say I hope that you folks learned a little bit something about that precious ball in the sky that gives us energy and life. We'll be starting in a couple seconds. Just wait for people to trickle in again. So be sure to get that code in. Okay, we'll give it 5, 4, 3, 2, 1, and let's go. Again, really quickly, after the countdown, there will be the rules. Only one prize per person today. And then here's again a quick reminder of what our prize is and what's at stake today. And then let's jump forward to our first question. First question is, how will the sun end its life? It won't die. It will become a black hole. It will explode into a supernova, or it will become a white dwarf. Yes, it will eventually turn into a white dwarf, but before that, it will turn into a massive red giant whose radius will engulf the Earth and Venus and Mercury, just to give you a scale of how big it will get. Let's take a look at the scoreboard. Oh, in first place, we have Arctic Ferret with Mystery Ant and Piranha. Our next question is, what did the ancient Romans call the sun? Did it call it Helios, Sol, Zeus, or Aurelius? Oh, I guess we don't have a third option here. I see some people got tripped up here. Helios is actually the Greek term for the sun, and Sol is where we get the term for solar. So let's see who remains standing on the leaderboard. Arctic Ferret still in the lead. What is the name of the particle that transports light out of the sun? Is it a photon? Is it a proton? Is it an electron, or is it a neutrino? Oh, perfect. Almost everyone got that correct. It is, in fact, a photon, which is super, super, super tiny. And it's super, super important. Oh, we don't see a lot of change in the scoreboard, but maybe that'll change with this final question coming up. Our last trivia question of the day. What percentage of the mass of the solar system is in the sun? Is it 50%? Is it 75%? Is it about 95%? Or is it 99.8%? Let's take a look. Get those last answer in. Yes, it is so massive that it takes up 99.8% of our solar system's mass. And let's look at our final leaderboard of the day. In third place, we have Shining Dragon. In second place, we have Mystery Ant. And in first place, we have Arctic Ferret. Maintained their lead the entire game. Well done. And of course, we will be in contact with you via email for those prizes. So keep an eye out for that. And thank you so much for joining us.
Thank you so much, Nicole, and thank you to everyone who joined today and to all my fellow panelists. I think we have like 2 minutes. If anybody has any burning astronomy questions about the sun or about anything else about astronomy, then feel free to ask. And I think there's a Q&A. We have from Science Rendezvous asking, can the public go to the observatory? Well, Mike, do you want to answer that one?
So that's kind of a little up in the air right now. So normally in the pre-COVID times, we would have public tours once a month, which were run by graduate students. And as things are opening up, we actually will hopefully start opening up the observatory again soon. So the best thing to do is to actually follow the Dunlap Institute on Facebook or on Twitter or Instagram, and usually things get announced there. So.
Thanks so much for that question. Are there any other questions for us? An anonymous attendee asks, will this be part of Doors Open Toronto? So I think, Mike, you can also answer that.
Yeah, so yes, we will actually be a part of Doors Open Toronto. Not only will have the balcony open, and then we'll also be streaming live views from the 8-inch and the 8 Alpha telescope. We'll have some telescopes on the balcony as well. So that is at the end of this month.
All right. Thank you all so much. That was super cool getting to do both the a little bit of learning a little bit more about the sun, which we see every day, but clearly there's so much more that I don't think, I mean, seeing the Kahoot answers, I think so much we still don't know, or the general public really doesn't know about something that's so important that we see all the time. So that was super cool. Thank you so much. And getting to see the observatory, which I guess because of COVID, we probably won't be able to see until much later. That was super cool. And seeing all the technology that are quite old, right? But we still have those same things that are able to give us so much information about something so far away. So that's super cool. I love that. Yeah, and I don't believe, I don't know if we have any questions, but we have an anonymous attendee who says that it was super fun. So I'm glad that was enjoyable for you all. Yeah, okay, so thank you again to the Astronomy and Astrophysics department for the presentation. This will conclude the Room 2 webinar, and we will now move into our closing remarks and invite you to join us at the keynote presentation on a different meeting link. Just before that, we do want to give a shout out to remind everyone that Science Rendezvous does have a Sci-Art gallery, the link of which I will include once again into the chat. And this is really just a virtual gallery where we've implemented a cool integration of both art and science together. So whenever you guys have the chance, definitely check it out. This will be on longer than the duration of Science Rendezvous too. So do look at it. But yeah, if we want to go on to the next slide, we have some examples of what you will see at the gallery. These are super cool images. So I do think definitely a cool thing to go and look for. Yeah, and before we do let you all go, we would also like to extend a huge welcome to the SR exec team. The dedication for this year's event, because without them, right, this year's virtual SR event would not have been possible. So here's a quick view of everybody who was part of the Science Rendezvous team, from the directors, the volunteering team, logistics, programming, promotion, finance and sponsorship, the Sci-Fair, the Sci-Art, as well as the SRHacks team. So some of us are here as well. So thank you all so much. And behalf of everybody, we also thank you all for attending. Okay, so thank you all for attending our 2022 virtual festival. We do hope you will be joining us again next year, and maybe that won't be virtual anymore, but we don't know. So for updates on our events, follow us on our social media at U of TSR. And we're done in this room, but we do have the registration link for the keynote speakers by Professor Dwayne Miller, who is the founder of Science Rendezvous, and he will be presenting to us his talk about what is life and can we measure it. So please join us in the new link for his presentation, and you can also find this link on our website in the program schedule, and we hope to see you there shortly. So Henry has also kindly placed in the chat the link for the new Zoom room. So if you want to all head over to there, but thank you all so much again. And it was lovely to host all of you, and have a lovely rest of your day. Thank you. All right, thank you so much. So everyone's just trickling out.